biotechnology, right?
Yeah, it's pretty much taking advantage of like the ability to make DNA and to read DNA, and a lot of automation and software to make all the stuff we used to do in the lab by hand in an industrial scale. So it's
Wow.
it's crazy.
Right. We are not admitting anyone.
Okay.
Yeah, just
So,
wait.
but they are here.
Yeah, they're here.
May I say hi?
Yeah. Janelle is here.
I'm not here. Because you have
Oh, he's invisible.
Oh,
background.
okay.
Background.
Janelle. Okay. Hi.
Ah,
Hi, Juan. Hi, John.
Hello, Kuya
John.
Janelle.
Hi, Ruby.
Hi, Ruby. It's been like, what, 10 years since I saw, no, 2
Oh,
years.
yeah.
Last week. I saw you in Cebu. Okay. But it's been 10 years since Boulder, so
I know, I miss Boulder a lot though.
I'll be in the other computer.
Okay, yeah.
Yeah.
Nice to see you.
I sent the question.
Okay. All right. So we are waiting for Cheryl. Cheryl Andam. She's the first speaker for today. Mon, do we with other sessions that you hosted, are there any? Can we like
ma'am.
5 minutes
The protocol is if the first speaker is not hasn't logged in after the 10-minute mark, we proceed with the next available speaker, ma'am.
Okay. All right.
did you say Jono? Because there is a Jono in the waiting
Oh
room.
no.
No, Janelle is not a speaker.
Janelle.
Ron.
Ron Panganiban is a speaker, ma'am.
Yeah, Ron Panganiban is a speaker.
All right. I'll admit him.
Oh, he.
Right. Cheryl Andam.
I'm looking for Andam, but there's no Andam in the waiting room.
Okay. Oh, there she is.
Ah, there she is.
Okay.
Just joined. And I've already have Sir Emzo's presentation loaded, so it's just ready to play.
Okay. Excellent. Okay. Let us know if we could start.
I think we can start, ma'am.
Okay. Can we just see Cheryl? Cheryl.
Hi.
Hi.
I was having problems. Sorry. With yeah.
Sorry, ma'am. It's my account. The university's settings for our Zoom account is quite stringent.
Yes, it is. It is.
Sorry about that.
It's
Sorry
okay.
about that. It's my fault. Sorry.
Okay.
So,
All right. So everyone is in for the first session. Do you think we can start now, Mon?
I'll start admitting people, ma'am.
Okay.
Science and technology and innovation, these are indispensable in the modern economy, in the modern world.
We held a meeting at the MSI, and we resolved among about 6 to 8 of us, local members, first to form PAASE Philippines so that we could continue to host meetings here, but also to go on a massive campaign to try to get support from the national government through the legislative and executive branches to invest more in science and technology. So It started with a white paper, a position paper that we co-authored among 6 of us, namely Ernie Pernia, Cesar Saloma, Redora Asansa, Toby Dairita Fataneo, and Alvin Colaba of Dalisay, and myself.
So PAASE historically, in the past 10, 20 years for sure, have been collaborating with our counterparts here in the Philippines at various universities. And a lot of these interactions probably started as peer-to-peer. But thankfully, we have the Department of Science and Technology here in the Philippines, and they've come up with some really helpful and very impactful programs that actually enable PAASE members that are based in the United States and other parts of the world to actually come here and do extensive research and academic partnerships and collaborations.
Well, I think that one of the good things that PAASE is doing is really trying to stay in touch with the researchers and the scientists here in the Philippines. So I think that is one of the good programs that they keep on supporting some of the initiatives of our researchers and scientists here in the country, trying to align also their assistance towards the plans of the Philippine government.
It's very important that the PAASE members and scientists based in the Philippines are continuously in contact, so we will know the needs of the Filipinos and opportunities that Filipinos can have to do science with their PAASE counterparts. So these interactions, such as the conference that we're holding and the annual conferences that we have.
The Philippines and the scientists there have very unique perspective that adds to discoveries and inventions, advancing in general the field.
These members, because they are part of an academy, it implies that they are productive. So they have publications, they have innovations, they have inventions, patents, and they have, they have an impact on society. Okay, so these are the people who can convince government, invest more in science and technology, and that was really the gist of our paper, our position paper in 2000. And it is Ernie Pernia who said that R&D by nature is expensive and it is a public good. So it is important for the government to take the lead in investing in R&D.
To increase the economic growth potential of the country, we cannot continue to just rely on traditional inputs into production as capital and labor. We also need to develop our expertise in science and technology and innovation so that we can come up with new systems, new ways of doing things.
I think that science, researchers, industry, and even the government is really like an ecosystem of knowledge. So I think it doesn't really matter if you're coming from a private institution or a public institution, such as a state university. I think we all have a role to play to improve the research capabilities and scientific knowledge here in the country. I think it is about time for us to realize that one of our roles as academics, as researchers, as scientists, is to actually contribute to that global body of knowledge.
And the role of this cooperation among these government agencies really is to be able to attract science and technology, global-based companies to set up shop here. And that is really key in terms of developing the science and technology innovation ecosystem in the country.
Particularly because there's that deep desire. And what PAASE does and can do more of is to promote this relationship between its members.
We really need to ratchet up the appreciation, the recognition of the importance of science and technology and innovation.
And we are all trying to attain what we call the common good, the greater, higher, long-term good of the Filipino people.
So good morning, everyone. So Mon, do you still have another video to play, or that's it?
That's it po, ma'am.
Okay. All right. So I'm going to share my screen. Okay. So good morning, everyone, and welcome to the 10th parallel session of the 40th PAASE anniversary and the 2020 APAMS Annual Scientific Meeting in the Philippines. So today we are all very excited to hear from our very young, talented, early career scientists who've been working so hard on their science in the field of chemistry and biochemistry and also on molecular biology. And today we have three different subsessions. First topic, we have the genomics and synthetic biology and metabolomics, followed by the biosystem interactions. And the third topic is the drug delivery and biomarkers. So what we are going to do with this session is that we are going to finish the first topic, and then after that we will have the question and answer, and then followed by the second topic, and then question and answer, and then the third topic, and then question and answer. That way, our speakers will not wait until the whole session will finish, and then the open forum will happen. And before anything else, so for the interest of everyone, I'm going to show you some. I'm going to show you some facts and figures about what the participants of this session. And so therefore, based on countries, there are 88% of the registrants coming from the Philippines. At the moment, we have about 146 registrants. 10% came from United States of America, and 2%, that is 1% in Korea, and then 1% in Japan. So what happens here next is, in the Philippines, so we are divided in three different regions, the Luzon, Visayas, and Mindanao. 47% of our participants came from Luzon, 35% came from Visayas, and 18% is from Mindanao. We also have the different sectors represented here. 59% comes from the education, which also comprised of graduate school and also research institution. We have 27% from the medical sector, meaning from hospitals and other medical sectors. We have 8% coming from the government, 2% coming from industry, and 4% comes from other sectors. And so I'm going to introduce to you our first speaker. Our first speaker is Dr. Cheryl Andam. She is an assistant professor of microbiology at the University of New Hampshire. She will be starting her new faculty positions, very exciting, at the State University of New York, or SUNY, at Albany this fall, September 2020. Her research focuses primarily on the microbial genomics and molecular evolution. She was trained at the University of the Philippines for her bachelor's degree, in Binghamton University, New York, for her master's. For her PhD, she finished from University of Connecticut and did her postdoctoral training at Cornell University and the Harvard School of Public Health. She was awarded a DOST Balik Scientist twice, in 2017 and 2018, and she's currently the recipient of the United States National Science Foundation's Faculty Early Career Development Program Award. So ladies and gentlemen, I would like to present to you Dr. Cheryl P. Andam for her talk entitled Causes and Consequences of Genome-wide Recombination in Bacterial Species.
Oh, Cheryl, I think you're still muted.
Mute, yes. So can everyone see my screen?
Yes.
Yes, yes. Okay. Well, good evening, good morning, good afternoon to everyone. It's currently 8 something p.m. here in upstate New York, where I am based. And I'd like to thank the PAASE and Dr. Dalisay for the invitation. It's it's an honor and a pleasure to see everyone tonight. Today, I'm going to talk about, one of the things that I'm very, very passionate about that I've been studying for the last more than 10 years. The genome-wide recombination in different bacterial species and what are the causes and consequences of recombination. And I'd like to start by mentioning these mythical creatures in Greek mythology that some of you probably have already, have studied in the past, have seen in pictures and books. This is a chimera, and it's a mythical creature made up of, the heads of 3 different animals. We have the serpent and the goat and the lion, and we all know that these are, they're mythical. They're not real. But I'm going to convince you today that these chimeras do exist in the microbial world, and they are quite a lot of them. Different species, different genera, and so on. So let me start by defining recombination, specifically homologous recombination. It refers to the genetic exchange of genetic material between different strands of DNA that contain long stretches of similar base sequences. So in this diagram, there is an incoming, a short fragment of an incoming DNA, DNA that enters the cell. It has 3 genes, gene A, gene B, and gene C. Now the host genome has the complete chromosome, but it also has those 3 genes, gene A, B, and C. But the gene B is a different version of the gene B in the incoming DNA. And if there is some, a substantial similarity in the sequences, in DNA sequences of the incoming DNA and the host genome, this could result to a double recombination. So basically replacing or switching between these strands, and this can result to a recombinant host genome that has the gene A, the original gene A, and the gene C, but in this case, a new version of the gene B that was initially present in the incoming DNA. And this process can result to a substantial genetic generation of substantial genetic diversity in a species, in a strain, in any kind of organism that can acquire DNA, incoming DNA from other sources. And so there are 2 ways in which these microbes, or in which recombination can result to genetic diversity within a cell. First, it can result to the replacement of genes in one lineage by homologs from another lineage. Sorry, I keep hearing that ping sounds from, I don't know, from the audience, I guess. Anyway, so recombination can result to the replacement of genes in one lineage by homologs from another lineage. So if you have an incoming DNA, in red here, it can simply replace that same DNA strand in the host genome. Or alternatively, recombination can also result to the addition of new genetic material. Here, there's a red DNA, we'll just call it by the color, red DNA inserting itself into the chromosome of the cell. And so both of these output, the replacement and addition of DNA, can happen as a result of recombination, and as I've said, can result to a substantial genetic diversity within a species. the current models of microbial recombination, for any viral, bacterial, archaeal species, there's this assumption that recombination is a homogeneous process across the species. In what I mean here is that if you look at one species, it will only has one, it can be characterized by a single pattern, characteristic, rate of recombination. And so this figure, this plot, sort of shows this this recombination as a homogeneous process where, and the y-axis is the recombination to mutation ratio, and these dots show the recombination rates for different species, Flavobacterium, Pelagibacter, and so on. And so when we calculate these recombination ratios, we use these numbers, these recombination characteristics of each species to understand, for downstream analyses, for applications, for example, in clinical settings. However, what's not often recognized and not often appreciated is that even within the same species, there can be substantial differences in recombination rates within among strains of that species, and these are indicated by these whiskers. And so this is what my lab studies. One of the things my lab studies is this variation in recombination characteristics within species and between species. And so this is just to give you an overview of the things that we study. I don't have a favorite organism. I study both antibiotic resistant and antibiotic producing bacteria, mainly Staphylococcus from humans and animals, Campylobacter, Salmonella, Streptococcus, and Streptomyces that naturally produce antibiotics. And so I'll just give you, for tonight, I'll just give you a few examples of these. One of the things that we study is Staphylococcus pseudointermedius. So those people who have pets, you've probably heard of this bacterium. It's a commensal and opportunistic pathogen of dogs and cats. We've been sampling Staphylococcus pseudointermedius in dogs and cats in the New England region in the northeast of the US. And we're finding, many methicillin resistant staph pseudointermedius, and in fact, Staphylococcus pseudointermedius is very, very closely related to Staphylococcus aureus, which infect humans. So almost every month we see methicillin resistant pseudointermedius in our data set. But not only that, we also see many strains of pseudointermedius that are multi-drug resistant, indicated in this plot. So many of them are methicillin resistant, but a substantial number of them are also carry resistance genes for different families of antibiotics. The next thing we did was we compared the Staphylococcus pseudointermedius that we sampled with the global population from North America, Europe, and Australasia, and we just look at their genome sequences. We found that, again, for each geographical population, there's a substantial variation in the polymorphic sites, and these are indicated by, the different geographical populations are indicated by these different colors. So they vary in their polymorphic sites, in their accessory genes per genome. Accessory genes are those genes that are present in some strains, but are absent in other strains. And also in the presence and presence of antibiotic resistance genes per genome. And in fact, it's quite striking to see how many resistance genes are present in each genome. There could be as many as 20 to 20 antibiotic resistance genes in a single strain. So again, we're talking about just one species, but each strain is very different from another. And we think that this variation, genomic variation, partly stems from the frequent recombination of pseudointermediates. So this is a phylogenetic network that shows the relationships of the global population of pseudointermediates their network-like structure indicates that there are the presence of the existence of recombination. But not only that, not only do they frequently recombine, but when we look at the 3 geographical populations, they also differ in the amount of acquired DNA per genome. So for example, so in this case, this is referred to as recombination coverage. In North America, 23% of the on average, 23% of the genome came from somewhere else. In Europe, around 19% of the genome has been acquired from other sources, and the same is true with Australasia. The rates of recombination also slightly vary among the 3 populations. And so again, I just want to emphasize, we're just talking about a single species from different geographical areas, and they differ in their genomic content and in their recombination rates. We also wanted to look at the genes that are frequently recombined among these 3 populations, and just looking at the numbers and the patterns of the dots indicate the different genes. We see that they vary among the 3 geographical populations, but there's one gene, CUP-B, that functions in copper export, and it is frequently recombining in all 3 geographical populations. We can see it in North America, in Europe, and Australasia. And this CUP-B gene, as I've said, it functions in copper export, and it enhances the survival of Staphylococcus aureus, which is closely related to pseudointermediates inside macrophages. And so that's a story for the Staphylococcus pseudointermediates. We also look at recombination in Salmonella enterica. This is a project that's recently published by my PhD student. For some of you who works on pathogens and infectious diseases, you'll know that Salmonella enterica can be classified into different subspecies, okay? And so we look at this matrix shows the genomic similarity among these different strains, and the more red the color is, that means the more genetically similar they are to each other. So we can distinguish the different subspecies from each other. So I just want to emphasize again, there are different subspecies in Salmonella enterica. And we know that each subspecies frequently recombine with each other, among the strains of a subspecies frequently recombine with other strains from the same subspecies. So all these enterica, all these diaryzonae, all these salame recombine with each other. But when we look at the overall recombination pattern within the species, we find that there are some subspecies that preferentially recombine with certain strains, as indicated by these gray lines. So the gray lines indicate the donor DNA and the recipient, the donor and the recipient partner of the recombination event. So the more arrows you see, that means the more recombinations that are happening between them. So for example, you have these light blue strains frequently recombining with the dark blue strains, and the pink strains recombining more often with these smaller subspecies. But then you see these purple strains, this is enterica, subspecies enterica, that do not, these strains do not recombine as often as other lineages or other subspecies. And so what we're seeing here is that recombination is not random. There's some biases that happen as a result of phylogenetic affinity, maybe the same environmental or ecological niche, the same geographical setting, and so on. And this has been referred to as highways of recombination and has been identified in Streptococcus pneumonia. And again, it's the same, very similar figure here. We have recombining partners within the same species, and these highly recombining strains act as a major reservoir of genetic diversity in the wider population. And this is important because rare genes that tend to get lost in the population because of recombination, they get to be spread around and disseminated among strains of the population. We also see, so all the things that I've shown you earlier, these, this is just recombination within a single species. We've also found, recombination in Streptomyces that are between different species. And what's really interesting is that, so in this figure, again, this is a phylogenetic network of around 130 different species of Streptomyces. We've identified ancestral populations, 7 ancestral lineages of these in this data set. And if we look closely, we see that there are some species that are made up of their DNA is made up of DNA from all from the different 7 anyway. So so example of this species is Spiroverticillatus. Okay, so these hyperrecombinants or frequently recombining strains we can consider them as hopeful monsters. This is a term in non-Darwinian evolution, which states that evolution can proceed in jumps brought about by sudden large-scale genomic changes. And we call them hopeful because these are dramatic changes that happen to the genome, which are often deleterious. But in rare cases, we see recombination events that can be highly adaptive and allows the recipient to exploit a new niche, and that's why it's called hopeful monsters. And this is clinically relevant because these hopeful monsters can become public health threats that, and their impact can be very hard to predict. Examples of these are, Streptococcus pneumonia that can acquire antibiotic resistance genes more often than others. This is a figure where we have the red, I'll just refer to them by color. We have the red genotypes, red strains and the green strains, but then you have these yellow strains that tend to be associated with higher levels of resistance to different antibiotics, indicate penicillin, erythromycin, and so on. This epidemic Klebsiella, this particular lineage ST258, which has resulted to multiple outbreaks since the 2000s in many hospitals in the US. In fact, it has emerged as a hybrid strain between 2 other strains. We'll just call it the red strain and the blue strain. The hybridization resulted to this new lineage ST258, and there was a second acquisition of DNA. It's a mobile genetic element that also contains virulence genes. And then there was another recombination that happened with the green strain. And so the strain that has been circulating in hospitals and causing, outbreaks in the US really came from emerge as a result of multiple recombination events that have happened in the past. Again, an excellent example of hopeful monster. And so we study not just the emergence and the epidemiology of antibiotic resistant strains, antibiotic resistant pathogens and their virulence, but we also ask questions related to that are more fundamental to microbiology especially. What I've shown you today is that bacteria, they may be clonal, they reproduce clonally, but strains of the same species are not genetically identical. And this begs the question, and I would like to leave this question to you for you to think about. Do prokaryotic species exist? And if they do, how do we define species boundaries? If these bacteria are constantly acquiring DNA from different origins, do we even call them by the name that we're used to, that we used to call them? Is salmonella and streptomyces and staphylococcus and so on. And so I'd like to leave this question for you to really to think about. And I'd like to finish this talk by acknowledging, my grad students, my collaborators, my funding agencies, and also, as Doralyn mentioned earlier, I would like to shamelessly mention that I'll be starting a new faculty position at the University of Albany, State University of New York. And if you have any questions, please feel free to send me an email. Thanks.
Thank you very much, Cheryl, for that very, very interesting talk about the amazing power of these microorganisms to steal or recombine the different DNA in that environment. So thank you for that. And there will be some questions later after this subtopic will be over. So thank you very much. So our next speaker, is Dr. Emzo de los Santos. He's a Filipino, but he obtained his Bachelor's of Science degree in Biological Engineering at MIT in 2009. He received his PhD in Bioengineering from Caltech under Professors Murray and Stefan Mayo, where he worked on engineering novel transcription factors. In 2015, he joined the Warwick Integrative Synthetic Biology, or what we call as WISB, Center as a postdoc, a research fellow under the supervision of Professor Greg Chalice to explore methods for the in silico analysis of biosynthetic gene clusters. He is currently a research career development fellow at the WISB, where he is pursuing computational and experimental techniques to facilitate the discovery and engineering of new biosynthetic pathways. So since Dr. De Los Santos is based in London, and it's around 1 a.m. in the morning in London, we have a pre-recorded presentation of his talk, but he's very willing to accept questions later that we could send it to him by email so that he can respond to that particular question later. So, ladies and gentlemen, here is the pre-recorded presentation of Dr. De Los Santos.
Hi everyone, good morning. My name is Emzo de los Santos and I'm a Research Career Development Fellow at the University of Warwick. First of all, I'd like to thank Doralyn for the chance to talk about my research, which is on the tools for analysis and engineering of biosynthetic gene clusters from people back home. So what's my research about? We know that microbes can be the source of interesting compounds. We see this in the pharmaceutical industry, where penicillin was from a fungus and it was the first antibiotic discovered. They also have applications in the agrochemical industry, and below are some examples of different compounds that they've isolated from microbes. And you see that they have a variety of applications as antimicrobials, antifungals, insecticides, and anti-cancer drugs. So this caused some excitement in the natural product research community, and there's been a long history of this grind and find approach, where you find a strain with interesting bioactivity, and then you do some analytical chemistry in order to identify the compound of interest. And once this occurs, you're able to isolate this compound and use it for the application that you produce. However, what's happened recently is this has stalled as the rate of rediscovery of already known compounds has increased, leading pharmaceutical and other companies to come away from this, from natural products. However, it's seen a resurgence of interest in recent years due in part to the to cheap DNA sequencing technology. What this allows us to do is it allows us to link different biosynthetic gene clusters, so what genes are actually producing the compound of interest, to the compounds. And this has shown great promise because we see that there are a lot of biosynthetic gene clusters in these microbes that code for compounds that we don't know about, or that can be potentially novel compounds. So the reason we're able to do this is because the genes that are responsible for the biosynthesis of a certain compound tend to be clustered together. Because this is the case, you can take a genome sequence and run it through biosynthetic gene cluster prediction software such as anti-smash, and this will give you a prediction of the biosynthetic potential of the different organisms. And once they saw that there was a lot of undiscovered potential new compounds out there, there's been a rush to sequence and to discover more natural products that are derived from these microbes. This, however, has been a double-edged sword as the amount of sequences that we had are steadily increasing. These figures are from 2 years old already, and you can see that there were 600 million whole genome sequences in the database and about 2 trillion or quadrillion bases. This has called for some people to ask for a sequencing moratorium until we can figure out how to actually analyze this data properly. If you look at it from the biosynthetic gene cluster perspective, there are two studies. One of them is the anti-smash database, which looked at about 24,000 representative bacterial genomes. And in this, we identified about 32,000 potential biosynthetic gene clusters. A more recent study that tried to look at the biosynthetic potential of all the publicly available genomes found about 1.2 million potential biosynthetic gene clusters. Now, obviously, this is a lot, and it's difficult to know which ones to look at or which ones to analyze. So you need some computational way to prioritize which biosynthetic gene clusters you analyze. So this is kind of my research interest the past few years, where you have this sort of natural products 2.0, where you tackle the big data age systematically, first by in silico screening biosynthetic gene clusters of interest, and then testing them in an in vitro setting before the more costly in vivo testing and scale up needs to be done. So I'm going to focus my talk on talking about the in silico and the in vitro part of this pipeline. So these are my current in silico projects. One is on genome mining tools, which I'll talk about a bit later. And the second is on some machine learning approaches to identify new types of biosynthetic gene clusters. And I just want to highlight that I've also done genome assemblies with a group in the Philippines based in UP Los Baños, and we've looked at some of their actinobacterial strain collection. And this is something that I hope to do with Doralyn as well in the future. So the two tools I want to talk to you about for mining potential biosynthetic gene clusters of interest are Bigscape and cluster tools. Bigscape is something that I worked on with a larger group that's based primarily in the Netherlands, and cluster tools is something that I developed as a postdoc in Greg Chalice's group. So what Bigscape does is a lot of these biosynthetic gene clusters tend to be fairly similar and have the same genes in them. So if you're able to align these gene clusters based on their PFAM domains, which are their functional domains, then you can group clusters that are similar together, and you're able to analyze the clusters as families instead of individually, and this helps a bit. So what this allows us to do is two things. You can compare it to known biosynthetic gene clusters. So this is a cluster from a strain that we analyzed in the Philippines, and you see that it's quite similar to a known biosynthetic gene cluster with a few differences. So we can be fairly confident that they're making the same compound. The other thing you can do with this is start to explore families that aren't attached to any known biosynthetic gene cluster, and that way you can look for a new compound. However, this approach can only take you so far, because even when you group them together in families, there's still a question of how do you choose which family is the target. So lucky for us, the number of sequences allows an alternate approach. As you've seen previously, the traditional genome mining approaches take a set of genome sequences, run them through biosynthetic gene cluster prediction software, and you come up with an idea of the biosynthetic potential of the organism. An alternate approach is if you focus on the chemistry or the biosynthesis involved with specific chemical motifs. So this is a study that we did a few years ago on the polyketide alkaloids, and as you can see that they're pretty structurally diverse, but then because of this moiety over here, they actually share some biosynthetic, they have some biosynthetic similarities, and they go through a common biosynthetic route, which involves two genes. One is an aminotransferase gene, and the other is what's called a polyketide synthase gene with a reductive chain release mechanism. So if you can have a functional search based on the biosynthesis, you're able to explore chemical motifs of interest. In order to enable this, I developed software that allows you to query a genome database for functional units or co-localization of functional units. So you can have, you can look at a specific genome sequence and look for areas that have either specific genes or genes that have specified domain architectures or specified sets of domains. So what this allows us to do is you can use kind of a proximal functional search. So it kind of inverts the traditional genome mining approach in its head, because now you start with a motif of interest and a biosynthetic hypothesis on how these compounds are made, and then you query a database using cluster tools, and you get an idea of the phylogenetic diversity of your chemical motif of interest. So I'm going to walk through an example of how we did this in practice. So this is a set of compounds known as class 1 HDAC histone deacetylase inhibitors from gram-negative bacteria. And these compounds are of interest because they've been shown to have some anti-cancer potential. So we are interested in looking for compounds of this class in bacterial genomes. So as you can see, there have pretty similar structures, but there's a variable region that changes between the known HDAC inhibitors in gram-negative bacteria. If you look at how they're synthesized, this carries over where you have a starter gene that loads some kind of starter unit onto one module. You have genes that are responsible for creating this fixed region I told you about. And finally, you have the variable region genes, which can change between the different subtypes, but they all share certain domains that you can look for. So with this in mind, we conducted a cluster tool search on a bacterial genome database, and we got a few hits. Further analysis of these hits showed us that a lot of them corresponded to known biosynthetic gene clusters, or were pretty similar to biosynthetic gene clusters that we already knew about or that were in the database. However, we noticed one of the hits from a Pseudomonas species that looked different from all of the other hits. So we took a closer look at the domain architecture of this, and we were able to predict that it was actually the biosynthetic gene cluster for one of the known histone deacetylase inhibitors, FR901375, whose biosynthetic gene cluster hadn't been reported yet. So I forgot to mention that this work is in collaboration with Xinyan Jiang, who is a postdoc in Greg's lab. So luckily for us that this strain was readily available, and we had access to this strain, so we were able to get hold of this strain, and we saw that indeed it produced the compound as we predicted. So this is just some LC-MS data, which shows the wild-type extract of the Pseudomonas strain compared to the standard of FR901375. We were also able to confirm its identity with high-resolution mass spectrometry. Furthermore, when we deleted one of the genes in the biosynthetic gene cluster that my program identified, we saw that the compound was abolished, further linking that this was the biosynthetic gene cluster that was responsible for the biosynthesis of the gene. So this is one example of how you're able to use this proximal search algorithms to find genes of interest. And we've done it a few times. And one of the things I want to point out is that this approach isn't limited to biosynthetic genes, so you can use it if you have a biosynthetic hypothesis on how a compound is synthesized, but you can also use other genes or other functional elements such as regulatory motifs and resistance motifs. And this program is freely available for download, and it's been downloaded and used a bunch of times. So this is something that you can use for genome mining. So this is kind of what the workflow we have right now for biosynthetic gene cluster prioritization, where you start with predicting what biosynthetic gene clusters you have or you want to analyze using a tool such as anti-smash. And then you use Bigscape to dereplicate and cluster together the biosynthetic gene clusters that you're interested in. And then once you have that, you can use this proximal search algorithms like cluster tools, using different things like the biosynthetic signatures of different chemical motifs, the potential for activation if you know that you can bioengineer a pathway by overexpressing an activator of interest. That's something that you might want to look at, or you can look for something like antibiotic resistance genes if you're trying to find compounds that have antimicrobial activities. And then there's also the practical side of things. Once you have these genomes, you want to look at strains that are actually available. Lucky for us, the Pseudomonas strain that we found was something that we had access to, but then if you don't have the source DNA, then there's no point in looking at those strains. So that's the first part of my talk, where I talked about the in silico screening of omics data. And the idea here is that you're able to use this in silico screening to identify interesting parts and clusters. However, even once you're able to identify a biosynthetic gene cluster of interest, it's sometimes still difficult to work with that biosynthetic gene cluster. Either the organism that it's from isn't genetically tractable, or it grows very slowly, or you want to modify the cluster in some other way. And for this, what I've been trying to work on is some sort of in vitro testing platform using what's called a transcription translation reaction. So why is this something that you want to do? Well, if you look at other engineering disciplines, they often have a test bed, which is a less complex system from the full system. It's really only in bioengineering where you try to put the entire genetic circuit, or you test the entire genetic circuit immediately in the entire organism before going, without going through intermediate testing bed. So on the left is a wind tunnel, which is used in aerospace engineering to model aircraft designs. And you have these in increasing complexity until you actually are able to build an airplane. And then the right is a breadboard. So this is something that allows you to prototype different circuit layouts to see that the circuit that you want to build actually works before you solder it into a more permanent state. So during my PhD, my supervisor was pushing the idea that you could use in vitro transcription translation systems as a biomolecular breadboard. So what these are their whole cell lysate. So you grow cells up to mid-log phase, and then you spin the cells down and you lyse them, and then you spin down the cellular debris, all the genetic material, and what you're left with is the transcriptional and translational elements of the cell, so the transcription translation machinery of the cell. You add to this an energy solution that has some things such as tRNAs, amino acids, and some buffers, and what you have is the TXDL reaction. So how do you actually run a TXDL reaction? What it is you have about a quarter DNA, 33% cell extract, and 42% master mix, which is kind of this buffer energy solution that I talked about. You generally split that up into 3 10 microliter reaction volumes. And if you have some sort of plasma DNA that expresses something that has a fluorescent readout, such as GFP, you can see it very clearly on a plate reader after a few hours. So I've been working in collaboration with Paul Fremont's group at Imperial and Simon Moore's group at Kent to try to push this technology for TXTL for natural products research. So TXTL has been pretty much widely used in E. coli, which is the common model organism for synthetic biology, but then not too much has been done yet on TXTL for natural products research. So one of the things we're focusing on is making Streptomyces-based transcription translation extract. Streptomyces is a genus of bacteria that's particularly enriched for these biosynthetic gene clusters, and there's evidence that having extract that is based on Streptomyces is more suitable for natural products research, because the proteins that, some of the proteins that are made by these biosynthetic gene clusters tend to be insoluble in E. coli, but are soluble in Streptomyces extract. And so the bottom figure is just a TXTL reaction that is a TXTL reaction in Streptomyces extract. So what I've done with my PhD student is we've tested the process of making this Streptomyces cell-free extract with other Streptomyces strains that are commonly used in heterologous expression. And what this gives us is kind of a genetic toolkit. So we're able to have some sort of idea of how strong different genes are expressed in these host strains if we try to transplant the biosynthetic gene cluster from one organism to one of these strains, which is commonly used for natural products research. The other thing that we're doing is we're trying to develop a genetic toolkit for Streptomyces using cell-free extracts. So in this case, we have a promoter sequence which we've modified to include an operator site for a transcriptional repressor. And then we use the transcription translation system to see if we're able to repress the production of a fluorescent protein and if we're able to induce it again. So the bottom is a heat map showing this, where we have a synthetic promoter that has a LAC-I binding site. And so if we titrate up the LAC-I repressor, you see that the fluorescence decreases, whereas if we add an inducer, the inducer of LAC-I, which is IPGG, you see that the fluorescence increases again. So in this way, we're able to have some level of genetic control of the elements of the biosynthetic gene cluster we're interested in a species that, that we want to use for natural products research. So what I've talked about today is kind of the research that I'm doing and that I'm interested in, to take an interesting biosynthetic gene cluster from an organism, analyze, find clusters of interest using in silico screening methods, and then first testing these elements in a transcription translation reaction before moving on to the more costly and more time-consuming in vivo testing and scale-up. So I'd like to thank all of you for your time and listening to me. And I'm not available for questions because it's late in the UK, but I've told Doralyn to hand you my contact information in case you want to send me a message about it. Thank you for your time, and I look forward to hearing from you. So this is, these are my collaborators at Warwick. I still work a lot with Greg, my postdoc supervisor. I work with Simon and Paul on the cell-free systems, and these are people that I've worked with on some of the computational tools and some of the strains. Patrick is my grad student, and these are my former people in my group. Thank you very much.
So thank you very much, Dr. De Los Santos, for that. Now, unfortunately, as what he was saying, he's not able to
answer questions later during the open forum. But feel free to send your questions in the chat box, and then we will email that to him. So our next speaker is the topic now will be the combination of the Dr. Andam and also Dr. De Los Santos work, which is a synthetic biology. So our next speaker is Dr. Joaquim Marques. He is the head of the Omics and Analytical Chemistry of Gingko Bioworks. So he is representing the industry sector at this topic. He received his bachelor's degree in chemistry from the University of Sao Paulo in Brazil, where he also pursued his PhD in organic chemistry, working on the chemistry and biosynthesis of natural products. So after completing his PhD, Dr. Marques moved to the United States, where he worked for Dr. Norman Lewis Lab, investigating the biosynthesis of medicinal natural products, and later with the engineering of plant metabolism. So after his postdoc fellowship, Dr. Marques moved to Boston to work at Gingko Bioworks, the Organism company, exploring the interface of chemistry and biology at the forefront of synthetic biology. So ladies and gentlemen, I would like to present to you Dr. Joaquim Marques for his talk entitled Engineering Biology on this industrial scale.
Thank you, Doralyn. Right. Let me share my screen. So I'd like to first thank everybody. Thank you, Doralyn, for the opportunity. It's a huge pleasure to share some of this experience with everyone here. So to start, there's a slide that some people use my company, and I think a lot of people will find it interesting, is if we look at a picture like this, and if you ask a normal person, let's say, what is the most powerful technology there, they'll probably point to the telephone or to the laptop. But I think the most powerful technology here is the plant. So I do think that biology is the most powerful technology available. It is a technology that's self-repairing, self-assembly, self-replicating, and it's completely renewable. Not only that, it's a technology that's able to work with nanoscale precision, moving basically single atoms, and it can also, on the other hand, work in continental scale, right? It's shaped the planet Earth and define the composition of our atmosphere. So I wanted to give a brief talk a little bit, and Doralyn already talked about it, but yeah, like she said, I came from Brazil, from Sao Paulo, and that's where my passion for biology started. We do have a lot of rainforests and a lot of biodiversity, and I was studying the chemistry of plants in Brazil, and I was always fascinated by how competent plants and microorganisms are to do very complex chemical reactions, which I was at the time trying to do using organic synthesis. After I finished my PhD working with natural products, I had a chance to go to Washington State, and where I worked, I met Doralyn, and we worked at Norm Lewis Lab. And that's a place where I was able to learn a lot more about the biochemistry, molecular biology, how to find out what are the genes involved in biosynthetic pathways that make all those chemicals that were so interesting to me. Eventually, I had the opportunity to engineer some organisms, mostly plants, but we also use microbes to express biosynthetic genes from plants. And then I was very fortunate. So it was a big change coming from Brazil to Pullman, and it was perhaps an even bigger change when I got a job at this company in Boston. So I moved across the United States to Boston to Gingko Bioworks, And that is really a whole quantum change in the scale of the work I was doing in a whole different approach. So Gingko is headquartered in Boston. We are one of the largest synthetic biology companies in the world. This is a picture of one of our labs, and we are working on basically our mission is to make biology easier to engineer. Like Dr. Enzo de los Santos said, it's not trivial. And we are trying to work to make that something that is more accessible and easy, to make that, that technology available at a scale that can have a real impact. And we do that by using a lot of high-tech, especially software automation and things like next-generation sequencing, mass spectrometry, and so on and so forth. Just to give a few examples, so Gingko is very agnostic in terms of which projects and what are the things we work on. Just as to give a few examples, one of the projects we're working on is engineering microbes to be used in agriculture. So Gingko and Bayer created a joint venture that is engineering microbes at Gingko to do things like improve nitrogen fixation for plants that don't usually have microbes associated with the root system, unlike, let's say, soy. We have projects trying to produce natural products like cannabinoids, which is a huge market now, especially now that in the US, cannabis is becoming legal. So we're trying to move cannabinoid production from plants to microbes where you can scale and work. It's more amenable. You don't really need lights. You can just have a fermenter and you can produce those compounds. Another field which is moving away, I guess, from small molecules is motif, where we're trying to produce different proteins, including proteins from animals to use to make food of the future, moving away from animal protein to still animal protein, but produced by microbes. And a final one, which is, I think, one very interesting field that we're working on now, is a partnership with another company here from Boston called Synlogic, where we're engineering microbes that people can eat as similar to probiotics. And these microbes will colonize your gut or will live for a certain period in your gut, and they will help with different problems that people might have. For instance, phenylketonuria is a very common problem where people are unable to process phenylalanine. And so if they have a bug in their gut that breaks down the phenylalanine for them, they are able to have a normal diet. And so this just shows how broad and the variety of things that you can do with biology. And I think this is one of the things that is interesting at Gingko. So Gingko was founded by this man, Tom Knight, and 4 of his students in MIT. And I think he had a very insightful career change in the 90s. He was a professor of electrical engineering at MIT. And in the 90s, he was like, I don't think computers and electrical engineering is going to continue to be the main thing. I think in the 21st century, the technology is going to be biology. And he shifted his career. And he's one of the founders of synthetic biology. And one of the main things is that biology is fundamentally programmable. So Tom Knight basically had this insight that it's basically code encoded in DNA, and you can, just like any computer program, you can program biology if you have the right technology to do that, right? And then him and his students created Gingko, and leveraging that insight, but also 2 facts that are very remarkable, which is the very fast drop in cost of DNA sequencing, and on the other hand of DNA synthesis, which is the synthetic part of synthetic biology, at least in our case. So we usually, it's not that the microbes are fully synthetic, but we use a lot of synthetic DNA. And this goes to another point that Dr. De Los Santos mentioned, which is how hard it is to test all of that knowledge that comes from sequencing DNA. And I think now with the DNA synthesis becoming cheaper, it's possible for us to basically explore a lot more and really get into the engineering part of tweaking that DNA synthesis and reprogramming biology. There you go. And so the way Gingko is organized is pretty much like an engineering discipline where you have a design, build, test cycle. This is how we work at Gingko. We call it the biological foundry. So we have the design group, which is a lot of people with computational biology background, and they are experts in doing things like mining genome databases, finding genes, and thinking of the strategy of how you're going to assemble those constructs, those genetic constructs, to get to your desired goal, be it what are the genes you need to, let's say, make a natural product. They do that mostly on the computer, and then we place orders for DNA that comes to Gingko. We also have a DNA synthesis group in-house, so we also make a lot of DNA. And then we have the build group where they basically assemble plasmids, larger constructs, or they integrate that DNA into the genome of different species, different microbes. And then it goes to test, which is where my group works, where basically we test all of those things, right? So we test things all the way from the individual enzymes that we order. So, and I'm going to talk a little more about that later, but we test thousands of genes before we figure out what are the best genes to put together into a biosynthetic pathway. And then we test a lot of different ways of integrating that pathway into the genome. There's a lot of mass spec involved. We have, because of the diversity of projects we have a very diverse portfolio of analytical capabilities, because we never know what the project is going to be, right? So we've worked with things that are like fragrances, so we need GC to analyze, and we work now with proteins, so we need different types of mass spec. And one crucial part, which is part of test, but we keep it slightly separated, is the fermentation, which is how these microbes are going to be deployed in the real world. So we do have a highly automated fermentation development where we can do process development, like all the parameters of fermentation, before we go to scale up and deploy those organisms in the field. So this is what the labs look like at Gingko. We do have a very open lab with a lot of the power and gas coming from the roof, and everything is kind of in rolling tables, because we are still figuring out what is the best way to do all these things, and we like to have a lab that we can rearrange depending on the demand and on the problem we're facing. Right now, we just went through a major remodeling of our labs, because we switched from a lot of those projects I mentioned to a lot of other projects that are related to COVID response. And so here you can see a few of the robots that we use. Like I said, we are heavily, we use a lot of automation and software to keep track of all the things we do. So basically the way things work at Gingko, we have program teams which are microbiologists and people who are specialists in, let's say, yeast or E. coli or any other organism or type of, let's say, terpene products. And the foundry is kind of like a factory that we can, basically they come up with ideas and the foundry executes all the research. Everything is connected through automation and software, so we're able to track every sample, every operation I do when I'm like preparing samples. Let's say you grow microbes from the moment the DNA enters a Gingko. So when design places an order for, let's say, a gene, that gene is logged on our software, and then when it arrives and it's built into a microorganism, we track everything that is inside that organism, all the samples that derive from that engineer organism, all the tests that we make on that organism. And so we are basically trying to learn as well as we can, and the best way is to keep track of everything we do through software. And also, because we are basically recording everything on software, we can use those software instructions to basically to do the automation part. So we record in a laboratory information management system, and all the operations are then automatically can be transferred to our automation. This is one robot that we use a lot every day. This was a major game changer in my life. So when I was in Pullman, I'd never seen a multi-channel pipette in my life, and I spent many, many hours pipetting samples to set up enzymatic reactions. And at Gingko, with the help of these, that was me. With the help of these robots, we're able to basically, took me, let's say, a year to test 20 enzymes at Washington State. At Gingko, we're able to test thousands of enzymes in a single week by using automation. That's one, the one I just showed from the video, that is a walk-up automation, so anybody uses that. And we also have other automated work cells, is how they're called, where you have basically a robotic arm with a lot of different devices around it. So this is used for more complex operations, like if you're gonna, for instance, try to do a enzyme library screen. So around here you have a place that can incubate your plates where your bacteria are growing. You can have a liquid handler where you can do the extraction, you can centrifuge, you can put it into a plate reader, and you can seal those plates and put the labels. And so this is a very powerful technology this was built by Gingko's in-house automation team. So basically, one of the results of that, on the right side of this slide, you're gonna have that first slide I showed a few slides ago, where you see the price of DNA sequencing, DNA synthesis, and this is Moore's law, which is the drop in price for processing power. And on the left side is something that we've been calling Knight's law because of Tom Knight, the founder. But it is basically an observation that we made when we look back at all that data we accumulated over the years. And this is basically the log plot of any operation basically that we do in the foundry and over time. So this is also following an exponential trend. So with time, we've been improving our automation and learning more about how to do all the different things that are involved in this process. And we've been doubling our efficiency. So this is also holds true if you do a per capita operation. So we're really getting more efficient, and it goes, everything follows that trend. So liquid transfers, strain tests, which is basically how we call any kind of like a LCMS run, for instance, or pieces of DNA ordered, strains created. All of those things are progressing exponentially, and that's what makes this it's this core to Ginkgo's business. So it's the idea that we're doing more and more and learning, and as we learn, we're able to do more and better. And so it's kind of like a virtuous cycle of learning and building biology. And so that's how we walk towards the goal of making biology easier to engineer. Just to have an idea here, this is Gingko right now. Because of the way we've been doing this, we have the largest database of enzyme activity data. So when you look at UniProt or a lot of other databases, we have, most of them don't have actual data that like activity proven. It's more annotation, in silico annotation. And in a single project at Gingko, we test 20,000 enzymes or even larger numbers. More recently, this is a little bit old. And so we're basically learning, we take enzyme sequences from databases. We're able to express them and test them at high throughput and learn how they actually work, what's the substrate, how they express, and apply that knowledge to future projects. So this is another fundamental thing about Gingko, which is we have the foundry doing all these experiments at industrial scale, generating knowledge and information about the genetic parts, so to speak, that we called code base. And the more code base we have, we're able to do more experiments and take on more projects, more complex projects, and so it goes. So that's kind of very fundamental to the business model of Gingko. Okay, so going back to the foundry, right? I want to talk about my group a little bit now. Specifically, so we are the Omics and Analytical Chemistry group. We're a very core group at Gingko because, as probably most of you know, all of the experiments, you know, you can clone and you can engineer microbes, but most of the readout of like, is this working? It's going to be some sort of analytical chemistry most of the time. And so we do use a lot of mass spec as the main technology because it's a very diverse and flexible technology. So we have these two basic pushes. One is for throughput, so trying to make things high throughput using automation and some clever ways of using our instruments. And flexibility, because we never know what's the next project that Gingko is going to try to work on. So just to give an example, I want to use the example of, okay, sorry. This is, before I get to my example, so this is basically how it works, right? People have an idea, they think of what is that construct of DNA, what are the basic parts that you're going to need to make something. Let's say a natural product, you're going to need cyclases, transaminases, whatever. There's promoters, there's how you're going to link all those things. The design people will basically think about all of those problems and order DNA pieces from synthetic DNA. We receive that, we put it in the microbes, and then we test them. So testing, at Gingko, we have next generation sequencing and a more chemistry base. So my group doesn't do, even though omics is in our name, we don't do transcriptomics or genomics. We have a next generation sequencing group that is more focused on that. And we are focused on proteomics, fluxomics, and metabolomics. So that is extremely complex, right? If for people who work on metabolomics or proteome, you know, there's like thousands of molecules, thousands of genes that can be up and down regulated, and flux and cycles of molecules. And so this is a global view from CAG. And to be honest, most of the work we do is not at this scale or at this level. So usually we start by focusing on a specific pathway. So let's say a client want to replace some expensive chemical that currently is extracted from an endangered species. This is not something we work right now, but when I was, as a postdoc, I worked at podophyllitoxin, which is an anti-cancer drug, and is extracted from plants that are going extinct. So if we can take all those genes, put it in a microbe, and replace that, that's a very interesting proposition. So if you think of like a generic pathway, the way this would work is like the people at design, they would look at that pathway and they would say, okay, let's say there's 3 enzymes. So we go on databases, we have internal databases and sequences that we generate in-house, but you basically get all the enzymes, get the promoters, you think of how they're gonna be connected, syntons, and then the first step usually is enzyme screening. And so we test each enzyme separately before we string them all together. Those are usually the higher throughput needs. So we use, this is an instrument which is a mass spec, a triple quad, which is a very common instrument. The nice thing about this instrument is that we actually have 4 pumps feeding into one mass spec, so you don't need to wait for column reequilibration or for those parts in your gradient, in your LC gradient that are not interested. So you can basically overlap your runs, and we typically run at, let's say, 1 minute per sample. So we're able to really test really large libraries, libraries of 50,000 samples sometimes that we can run in a couple of days. I would say that as global omics at Gingko now, we're able to run something around 50,000 samples in a month. That includes this instruments, but also GC, proteomics, and other things. And we don't have a super large fleet of instruments. We have now 12 mass specs. And so it's a lot of samples running and not a lot of infrastructure, which goes to that high efficiency of these processes. We're always trying to increase the throughput. This is just one higher throughput, which is similar to MALDI, but is a laser diode thermal desorption. So the laser comes from behind the plate, heats up the plate, your thing gets into the gas phase, ionizes. And this is something that we've used to test larger libraries. So this will take a few seconds per sample. So we've been able to run enzyme screens of 20,000 enzymes on this instrument. And that's how we generate a lot of data. Usually, most regular biotech, they're going to use the known enzyme. And at Gingko, because of a lot of this computational knowledge and the ability to test a huge variety of enzymes, we do a very broad sampling of the design space, so to speak. So even if we're working on a biosynthetic pathway from a plant, we pull genes from all kinds of organisms, from microbes, from mammals. It doesn't matter. If it looks like that might be a gene that does that, we basically sample broadly from that design space, and we are able to get huge increases. So in this scale is a log scale, and these cluster here where the line crosses the samples is where you have your positive control, initial positive control, and you can see that we are able to improve dramatically, find enzymes that are much, much better than the starting point in your project. And this design space that we're able to explore allows us to find what, usually if you're moving closely to what you might find some local maxima that is not necessarily the global maximum. And when we spread out the diversity that we're looking at, we're able to find global maximum and get huge gains. After we have the best enzymes, we string them together, and then we start looking at pathway and doing pathway optimization, so adjusting the levels of different proteins, the expression and the host, substrate availability and things like that. And this is where we start to do broader testing, looking at flux, using isotope label tracing experiments in some cases, and we optimize those pathways. And then, to be honest, the real hardcore untargeted global metabolomics and proteomics is something that we use more when the project's not really working as you expected. And this is very common because biology is extremely complex and we're just beginning to learn how to engineer it. And so a lot of times you put great genes all together, they're all being expressed, but you don't have your products. And that's when we take a step back and do global metabolomics, where we're trying to look at all the different compounds that are being made, what are the imbalances, and we do use, we have access to some high Orbitrap, which have very high mass accuracy and allow us to find unknowns or do a truly untargeted experiments and troubleshoot. And sometimes you have to engineer things that are not even closely related to your biosynthetic pathway. We use a lot of tools, bioinformatics to slice through that metabolomics or proteomics data and to find the answers to drive our projects forward. This is basically to give an idea. So we do have global proteomics is still slow. We have regular LCs. We have that instrument, which is triple quad targeted metabolomics, where we're able to run tens of thousands of samples. The LDTD is expanding. We're expanding to MALDI and other technologies. And this is kind of like where we really troubleshoot all that synthetic biology and all that bioengineering and inform projects so they can progress and we can basically deliver products to market. This is obviously a very huge group effort. I think one of the things I like most about working at Gingko is that you're going to have automation engineers, software programmers, biologists of all backgrounds, people who work with plants, microbes, marine, people who have background in chemistry like myself. And that's what it takes to build such, to basically take on such a huge challenge. And I lost track of my time, but I would be happy to take any questions in the time we have left. So, yeah, thank you very much.
All right. Thank you very much, Joaquim, for that exciting, for sharing your exciting work at Gingko. So you will see the different types of technologies used in biology and chemistry to come up with some products that that could provide different types of platforms for fertilizers, food, or for medicine. So at this point, I would like to call on Dr. Cheryl Andam and Joaquim for the open forum. So for the first one, we have here a question for Dr. Cheryl. Cheryl, are you there?
Yes, I am.
Okay, so we have here a question: how is recombination related with horizontal gene transfer? What is their collective role regarding the diversification of bacteria or of a particular species? And second, geographical ecological barriers influence recombination of identical strain in disparate sites. However, for similar strains occurring in the same geographical site, what could be the factors that influence recombination or interspecies genomic variation? So go ahead and
yes, thanks for the question, and thanks to Dr. Ma'am Asuncion Raimundo for her question as well in the chat room. So recombination is really a process whereby there's a switching, replacement of similar DNA sequences. With horizontal gene transfer, you're talking about the acquisition of often new DNA, new genes, and then integration into the chromosome. And one of the processes by which these new genes are integrated into chromosome is through recombination process. And there are multiple different, kinds of recombination. I just talked about homologous recombination, and there's also illegitimate or non-homologous recombination, and everything in between. And then you have the mobile genetic elements as well. So, these are like multiple different processes. Most people tend to use the same these different terms in the same sentence, in the same concepts, but they are very different. In terms of diversification of microbes, so we'll just talk about bacteria, archaea, even viruses. There are two sides of the debate. First is that horizontal gene transfer, including recombination, can result in the diversification of any species because it adds new DNA into the chromosome. And once that DNA is integrated, if it's adaptive, it can be vertically inherited, and it can lead to the formation of new lineages, new species, and so on. But at the same time, recombination and gene transfer, if these processes occur more often among members of the same species, it becomes a force of cohesion. Instead of making two organisms diverge because they're constantly recombining with each other, they become more and more similar. So they become similar not because they evolve from the same ancestor, but because they are exchanging genes with each other. So it's a force of diversification, and it's also a force of cohesion. And you're right, in the second question, there are geographical, ecological, phylogenetic barriers, genetic barriers such as restriction modification systems in the cell. We have found evidence for organisms. So I'm also doing microbiome, metagenomic sequencing, and so on, but looking at the population level, strains of the same species. And what we're finding is that strains of the same species that live in the same environment, in the same host, for example, in the same nasal passage, and so on, we're finding differences in recombination rates as well. And this might be indicative of what we call cryptic ecological niches. These are very small environments in the bigger environment that we're not familiar with, that we have not identified. And so we call them cryptic niches. And there are many examples of those where, they do not recombine in the environment, even though they're coexisting with each other. But then you bring these two strains in the lab, and they do recombine, and vice versa. And so there might be something, and the thing is, we cannot identify all ecological factors in the environment. And so that might be, those are things that can influence or can act as barriers.
Okay, so thank you very much, Dr. Andam. And what we are seeing here, that these microbes constantly are very active in just doing their interchange of DNA whenever they are, whether they are in the same geographical site or different geographical site, they keep on actively sharing and recombining their DNA. So that's one amazing thing that we found with microbes. And I was really excited about the work that you are doing, especially with microbial ecology in different sites in North America, and I'm looking forward to collaborating with you in some aspect of our work here in the Philippines. So thank you, Dr. Andam.
Can I ask a question for Dr. Andam?
Yeah. Please call me Cheryl, by the way, to everyone.
Okay. Cheryl, I have a question. You were showing like a lot of recombination, and usually you have kind of like a source and a destination for the elements that are recombined. How do you determine where's the source and where's the destination? Like, how do you know that it came from here to there, not the other way around?
Yeah. So, like any genomic analyses, a lot of bioinformatics software that we use, it's mostly, comparing these sequences. So we have some tools such as Bayesian methods in phylogenetics, where we can compare, what is the probability of, this particular DNA coming from this particular, different groups of microbes, or whether that particular DNA came from your data set that you're analyzing or from somewhere else that are not included in your data set. And so
like if you're in your file, like the neighboring species
Yeah.
don't have it, and that's the only one, you're like, that must
Yeah.
be coming from somewhere
Yes.
else.
Yes. And so, different stats that go with that. And then we also look for markers such as, are there, mobile genetic elements flanking these, this specific gene? Are there, plasmids and so prophages and integrative elements and so on. So those can be used as other markers as well.
Cool. Thank you.
Okay, so thank you very much, Cheryl. So we move on with the question for Joaquim. So we have two questions. So one is, if you bioengineer a bacteria for a purpose to produce food, fertilizer, or drug, how stable is the bioengineered bacteria that it could sustain its function? And then the second question is, how does Gingko Bioworks respond to COVID-19 challenge in finding cure and vaccines? Has the epidemic shifted the priority programs in Gingko Bioworks?
Yeah, those are great questions, actually. So the first question is, it's actually a huge challenge that all kind of like biological engineering faces, because when you take let's say E. coli, and you force it to produce huge amounts of this useless compound, I mean useless for the E. coli and not useless for us, there's a huge selective pressure for the microbe to kind of lose that function, right? So there's different strategies. If you're doing things in a fermentation, basically you control the population that starts, and you can use certain selection markers. So we would definitely avoid things like antibiotics, but you can do auxotrophy. So like it only works if you provide some sort of supplement, and that's associated with the function that you want. So if it loses the function, it no longer is viable, and then it just dies out. That is a bigger problem in the field. So I think for instance, for the fertilizer application, let's say if you're trying to apply this in a farm, that's much more challenging. And people have similar approaches where if the bacteria loses the parts that you're interested in that give it the function, it loses viability. But it's not an easy to solve problem, and especially in fermentation is very common. If you have like long fermentative processes, as time goes by, basically bacteria are very good at escaping, basically tricking the system to stop producing that costly product that you want. And we try to use all of the strategies. We try to find locations in the genome that are more stable, but it's not a solved problem by any means. The second question, I think it's even more interesting, and it's something that made me very proud working at Gingko. So like I said before, we have like a huge diversity of projects. And when COVID happened, we were shocked like everybody. We basically paused all of our projects except for certain projects like the Synlogic projects, which are therapies that are, there's patients waiting for those products. So we kept those going as essential. But we took advantage of those two things that I mentioned, which is Ginkgo's flexibility. So we're trying to bake a biological foundry that is flexible and that can work on many different problems. And the fact that our lab space is also flexible, so we can rearrange robots and bring new instruments and take out old instruments. And we started, so the first action that happened, and it's something that made me very proud, was the founders, basically they committed $25 million to fighting COVID. So we basically made an open call for, and it's still open, for any academic lab or industry or group that is working on some aspect to fight COVID. Basically, we gave $25 million worth of access to our lab to accelerate those projects and to help with that. So that kind of led to several projects that are now ongoing. So they go from simple things like I mentioned, we have DNA synthesis in the lab. So we're basically making synthetic DNA constructs for research purposes. So like plasmids that express different parts of the coronavirus. So people who are trying to develop vaccines or drugs can basically get those plasmids ready to express and accelerate the research. And that's something that again, for anybody listening, you can go to AdGene and order those plasmids and those constructs that were manufactured at Gingko. We have a partnership, for instance, with Moderna, looking at vaccines. So Moderna, a lot of you might know, they're making this RNA-based vaccine, and one of the problems that is all vaccines are going to face is how to scale up. So we're not actually doing any kind of like optimization of the vaccine itself, but we're using our fermentation, high throughput fermentation and testing capabilities to basically optimize their fermentative process, increase the yields, and allow Moderna to basically produce more vaccines faster using our technology. We're also working on, so we have a pipeline that is very similar to the pipeline that Enzo was talking about for biosynthetic gene cluster for drug discovery. We had one gene cluster that was worked on a while back that showed some activity that actually didn't go anywhere, but we basically engineered a microbe to produce that natural product, and we sent that to partners to test as drugs. And we're also using all this knowledge on how to basically express proteins and produce huge varieties to produce both antigens for developing of testing kits, like for lateral flow assays where you need antigens, and to provide for people who are developing similar things like antibodies and vaccines. So it's been a major shift in our priority. And now basically we are trying to bring back the projects that paused when coronavirus starts, but definitely COVID is the #1 priority right now at Gingko. And we're basically slowly expanding our capacity to keep the COVID work while we start slowly to boot up other projects.
So thank you very much, Joaquim, for answering that question. So I think at this point, can we take a picture of everyone? So Mon, can you take a photo of everyone before we move on with the next topic? So Mon, can you say just
Sure,
one?
ma'am. Okay po. May I request everyone to please turn on their webcams? Okay, we are 55 strong, so this will take a while.
Yes.
Okay. Let's give everyone 5 more seconds to make everyone decent. Okay. All right. Thank you so much. I'm now taking pictures of the first screen. All right. Okay. And 1, 2, 3, please smile. Okay, there you go. That's done with the first screen. Please bear with me. Moving on to the second screen, please turn on your webcams. All right. Okay. Okay. Seems like no one wants to be taken a photo of for this. Anyway, I'll take a picture. Smile. Okay. And one more This is the final screen. Everybody, please turn on the webcams. 1, 2, 3, and smile. Okay, done na po, ma'am. Thank you.
Thank you very much, Mon. So thank you very much again, Cheryl and Joaquim, for sharing your interesting work in that drives the not new knowledge and also industry and application of science for the greater good of the community. So thank you very much. So at this point, we will start with the second topic, and this is with the biosystems interactions. So we have two young, talented, and early career scientists that we will be talking, or that will provide their that will give a talk about what they are about their work. So the first speaker under the biosystems interaction is Dr. Ronald Allan Panganiban. Dr. Panganiban is a research specialist at the Molecular Integrative Physiological Sciences in the Department of Environmental Health at Harvard T.H. Chan School of Public Health in Boston, Massachusetts. He was born and raised in Luksuhin, Ibaan, Batangas, where he attended his elementary and secondary schools. He graduated from University of the Philippines, Diliman, with a bachelor's degree in molecular biology and biotechnology. And in 2007, Ronald moved to the United States to pursue his PhD in molecular and cell biology at the Uniformed Services University of the Health Sciences in Bethesda, Maryland, where he studied the molecular mechanism of cell death in lung cells exposed to ionizing radiation. So he is currently pursuing his scientific training as a postdoc at the Harvard Chan School, where he was among the first to apply genome-wide CRISPR screens in metal toxicology, as well as among the first to describe functional role of GSD-MB gene, which he's going to talk about this morning. So ladies and gentlemen, please, I would like to present to you our presenter for the biosystems interaction, Dr. Ronald Allan Panganiban, for his talk on the functional role of Gasdermin B in asthma.
Thank you, Doralyn. I'm trying to share my screen, but for some reason it wouldn't let me.
How about now, sir? I made you into a co-host, so you should be able to share your screen.
It's requiring me a password. I'm sorry about that.
How about now, sir?
It says you cannot share your computer sound while other participants are screen sharing.
How about now, sir?
It wouldn't still let me.
Okay, so Mon, can we move on with Dr. Solian, and while you and Roland are fixing some technical glitch here. Would that be
Sure.
okay?
Yes, ma'am.
Okay. So, all right. So, so we move on with our next presenter, but we will go back to Dr. Panganiban's talk late after Dr. Sulyan's talk is over. So our speaker is Dr. Ruby Sulyan. She is a biophysical chemist with research interests in microbial nanoscopy, biofouling, and biomaterials. She obtained her Bachelor's of Science in Chemistry at University of the Philippines, Diliman, and her PhD at the Biointerfaces Group of Gilbert Walker at University of Toronto. While a PhD student, she spent a year at the Stacey Institute working on model membranes with Sean Zhou. Then she joined the single molecule biophysics lab of Tom Perkins at University of Colorado Boulder in the United States. In 2013, she started studying nanoscale properties of bacterial pathogens in the group of Dufresne in Belgium as a Marcel Diner Fellow. And in 2015, she moved to Germany as an Alexander von Humboldt postdoc fellow at Max Planck Institute of Colloids and Interfaces in the group of Kirsten Blank. So Dr. Ruby Sulyan is currently an assistant professor at the Department of Physical and Environmental Sciences at University of Toronto, Scarborough, and Department of Chemistry, University of Toronto, Canada. So ladies and gentlemen, I would like to present to you Dr. Ruby May Sulyan for her talk entitled Quantitative Imaging of Biofilms and Nanomaterial-based Antimicrobials. So Ruby?
Okay, yeah. So first, I'd like to thank Doralyn for this really great opportunity to tell you about the research that we do in our group. So in Brotto, so by the way, yeah, I am this is my first time presenting in an online platform. I only meet with the students via Zoom. So this is like exciting for me. So in a broader sense, our group is interested in the nanoscale interplay of bacteria and the surface. We all know that when bacteria adheres to a surface, that surface could be living, such as the host tissue, or non-living, such as a medical implant. So I'm not going to talk to you about the bacteria surface interactions today, but well, this part of our work focuses mainly on initial bacterial adhesion. We are actually also interested when this initial adhesion escalates into a more, I would say, complex bacterial lifestyle, which is the biofilm. And these two are, I would say, are the more mechanistic efforts in our group. And in parallel, to complement this more mechanism aspect, we are developing a multifunctional antimicrobial that is nanomaterial-based. And the goal here is that these multifunctional antimicrobials would be able to target bacterial adhesion as well as biofilm formation. And so before I get carried over, I'd like to first, oh, by the way, this is my focus today. So not the first two, but more the multifunctional antimicrobials. So before that, I'd like to thank these postdocs and grad students whom I would say did most of the heavy lifting. And in particular, I will talk mostly of the work by Nesha and a bit about the work that Christian did in the lab. Okay. So this is a headline from CNN that caught my attention late last year. So as you can see here, this is fairly recent, November 2019. So this headline, if you read it, every 15 minutes, someone in the US dies of a drug-resistant superbug. So this seems fairly dramatic, but then there is some truth to it. And you can kind of already get that sense from the first three speakers. In particular, the first two speakers that talk about antibacterial resistance in a way, and also like the role of microbes in contributing to that. So let me just, here. Okay, So that's for the I put it here, superbug that has learned to outsmart our most sophisticated antibiotics. And so because of that, there is a global effort in search for new antibiotics with a much more effective and novel targets. And I'd like to point out that Doralyn here, I recently came across her work that where they were able to discover new antibiotics from the marine sediments. So the point here is that many groups all over the world are actually working towards raising the microbes in developing antimicrobial. And that is such an important, I would say, problem globally. And hopefully this 10 years challenge that is circulating in Twitter we will tell you why it is such a problem. So what you see here in this image to your left is antibiotic susceptibility test where you still see this zone of inhibition or this halo around the drug. So back in 2009, bacteria in this particular petri dish, they are still susceptible to antibiotic. 10 years later, this halo or zone of inhibition disappears. So indeed, the rate at which the microbes develop resistance is far more is faster than we can develop antibiotics ourselves. And so we saw this 10 years challenge, and we kind of it prompted the group how can we contribute to this ongoing problem. And so our attempt here is to develop alternative therapeutic approaches that are not solely based on antibiotics. And the way we would go about that is to use a nanomaterial-based. So in a way, using some nanotechnology. And the idea behind using a nanomaterial-based antimicrobial is we want to expose the bacteria to multiple killing mechanisms. So in other words, we want to treat bacteria with multiple biocidal mechanisms in the hopes that because they are simultaneously exposed to different ways to kill them, it may be difficult for them to develop resistance. And that is what I would like to talk to you about today. So this is a work that Nesha in the group led, we nanomaterial-based antimicrobial. And I wanted to single this out, and I wanted to mention that this was just first published today. And I'm very happy that I'm able to present it in the PAASE conference. So with that, I'm not gonna take longer. Let me first tell you about the ideal properties of an antimicrobial. So as I already initially alluded to, an ideal antimicrobial, it should have multiple antimicrobial mechanisms. So again, different ways to kill bacteria. And one very obvious way to do that is, of course, having a bactericidal activity. So a good example of that are your antimicrobial peptides, which exhibits intrinsic lytic activity. So lytic meaning they can pierce into the membrane, they can damage the membrane, they can destabilize the membrane. But in addition, an ideal antimicrobial, it could also kill bacteria by serving as a nanocarrier for drug delivery. So I could, for example, take the antibiotics that Doralyn's group discovered and Janelle's as well and put it in our nano platform with the hope that in addition to, let's say, intrinsic lytic activity, we also have an added way to kill the bacteria. So using antibiotics, using drugs, using other antimicrobials. Another way to kill bacteria or ideal property of an antimicrobial is the ease in which to functionalize its surface. So what do I mean by that? So imagine you have an antimicrobial where you can easily functionalize its surface. You can confer many different functionalities. So one very important one is specific targeting. And I kind of remember Joaquim earlier, he talked about like precision. So that is one thing that's very important when we want to deal with this antimicrobial resistance. And another, I would say, advantage of having the ability to functionalize our surface is an extra functionality. So you can imagine here, if I put, for example, polymers, and I'm actually looking forward to a talk like after this, that talks about raft polymerization. So you can then think of like adding lytic polymers with antibacterial capacity and put them all in one platform, then you can achieve your multiple antimicrobial mechanism. But that is such an ideal property. But on top of that, your ideal antimicrobial should also be stimuli responsive, meaning we should have a way to control when do we want to release that drug, when do we want to enact the lytic activity. So in other words, we should be able to have externally triggered bactericidal activities. So these two are already very good, but still not enough. We want it to be, of course, biocompatible. So killing the bacteria is one thing, but we also don't want to kill the healthy cells. So we want to make sure that our antimicrobial will specifically target the pathogen and not the healthy mammalian cell. And of course, combining everything, we want also the antimicrobial to have a flexible platform. And what do I mean by this in particular in our study is we want it to be anti-fouling. So it has some anti-adhesion property, and of course, the anti-biofilm or antibacterial property. So these four, we just like put it together based on what we read from literature. And this is, by the way, the hope in developing this antimicrobial platform. So how did we do it? So our approach is very obvious. Number one, we started with a biocompatible platform. And this biocompatible platform is your polydopamine nanoparticle. So I will show you in the next slide how we form this polydopamine nanoparticle. But for now, we all know that dopamine is a neurotransmitter that's already found inside the body. And because of that, we don't need to worry about potential cytotoxicity. But then polydopamine on its own, it's actually non-intrinsic. So we want to confer intrinsic, it's not lytic or it's not bactericidal. So we want to confirm bactericidal property to our polydopamine nanoparticle. And the way that we do that is we make use of this antimicrobial peptide, which is CWR11. So we were not the ones who engineered this peptide. This is from a group that was published in 2013. So this CWR11, it's an engineered peptide that's rich in arginine and tryptophan, as you can see in the sequence that I wrote here. So if you count, there are about 5 arginine residues. So what it does, it develops electrostatic or interaction with the bacterial membrane, which leads to its lytic activity, and also the or specific targeting. And the W here, the tryptophan residues, it actually inserts into the membrane. So in a way, this peptide kind of has a dual function. It can target the bacterial membrane, and at the same time, it could also kill it. So we were very happy when we found this peptide. So back to our platform, we have this polydopamine nanoparticle. We decorate it with peptide. So it seems like it's a good platform to start with, but the thing that's missing here is how can we make it stimuli responsive? The good thing with the polydopamine that I did not mention yet is that polydopamine nanoparticle actually exhibits some photothermal capability, meaning we can shine laser to it, near-infrared laser, and then it heats up. And so on top of the antimicrobial peptide, we combined it with photothermal induced or laser induced photothermal heating. So this is like our platform or our approach in developing this antimicrobial platform. So just to sum up, we are combining laser induced photothermal heating with the lytic properties and targeting properties of the antimicrobial peptide. Okay, so first things first, can we actually synthesize this polydopamine nanoparticle? So turns out this polydopamine nanoparticle is, I would say, fairly established. Not so much, maybe in the past 2007. So like it's relatively new, but many people are working towards forming dopamine films and polydopamine nanoparticle because it is such a versatile nanomaterial. And so the way to form this nanoparticle is very easy. You just need to auto-oxidize or polymerize dopamine monomers under basic condition. So you don't need any fancy equipment. All you need is a vial. Throw your dopamine monomers, comes in a powder, put it in a basic condition, and lo and behold, you form your polydopamine nanoparticle. And so we monitored the growth of this polydopamine nanoparticle via DLS, or dynamic light scattering. So you can see here in the plot of diameter as a function of time of reaction, you can see that within 1 hour of the reaction, this nanoparticle already starts to form. So if you look at that, the diameter, it's about 45 nanometer, and it continues to increase in diameter as the reaction continues. And so we know that we are able to form these nanoparticles in the ideal size range. I said ideal because we don't want our nanoparticles to be bigger than our bacteria. We want it to be smaller than the bacteria because we want to target as many nanoparticles onto the surface of the bacteria. So this is one way to characterize it, monitoring its growth, looking at its diameter. We could also visualize it, and we've done so using TEM as well as AFM. So what this TEM and AFM, it confirms that we are indeed forming this spherical shape nanoparticles, in particular polydopamine nanoparticles. So that's good for the PDNP. So the next step, as I mentioned to you earlier, is to add the antimicrobial peptide to come up with our PDNP CWR11 or the antimicrobial peptide bound to PDNP. And so here I am, you are looking at AFM topography images. Okay. So topography images of the bare nanoparticle, so meaning no peptide, as well as in the presence of peptide. So I don't know how much can you see, but then if you actually squint, or maybe you don't need to squint, you can already see this like tiny nanoscale protrusions on the surface of the PDNP. So that's already a good, I would say, indication that we are indeed immobilizing this peptide onto the surface. And if I do a distribution of the sizes, there is indeed an increase in size or in diameter from 87 nanometers for the bare PDNP to 92 nanometers for in the presence of peptide. So taking this together, this provides us a first line of evidence that we have successfully grafted the antimicrobial peptide onto our nanoparticle. And to further verify or confirm that we are indeed grafted the peptide successfully, we also measured the surface charge or the zeta potential. And here you can see a dramatic increase from a negative zeta potential, so negative 33 to positive 33 in the presence of the peptide. So blue here is just bare PDNP nanoparticle. The red here is when we add the peptide. And this is, so if you have a successful immobilization of a peptide, you will indeed, you should observe an increase in the zeta potential because the net charge of this CWR11 is actually positive 6 at pH 7.5. And so taken together, the change in the zeta potential expectedly, as well as the sizes, confirms that we have this peptide bound to the polydopamine nanoparticle. Okay, so now that we are in a way comfortable and confident that we have formed the PDNP CWR11 accordingly, the next thing to show is the intrinsic photothermal capability of the bare PDNP. So I mentioned to you earlier, we can use this laser-induced photothermal heating to kill the bacteria. And so we first monitor if we can observe this in our hands. So we use a thermal imager so to monitor the change in solution temperature as a function of the dopamine concentration, nanoparticle concentration. So you can see here, so if it's not very clear here, you can see we are looking at 0 nanomolar of the nanoparticle, 0.5, 0.25, and 1. So for without the nanoparticle, this black line here, you don't see a change in solution temperature. But then as we increase the polydopamine nanoparticle concentration, the temperature also increases. So in particular, I'd like to point your attention that at 1 nanomolar PDNP, the temperature is about or close to 50 degrees. And by the way, the temperature needed to kill E. coli is about 47, 48 degrees, okay? Okay, so the next thing we did is to test its efficiency towards a drug-resistant bacteria, tetracycline-resistant E. coli, and this is what we got. So here is a plot of the bacterial growth as a function of the nanoparticle concentration, and you can see that the number of the viable bacteria decreases in a sigmoidal manner. If I fit this curve here with a Gompertz equation, I would get a value in microbiology, which we call as the MIC, or the minimum inhibitory concentration, where we don't observe bacterial growth anymore. And so here for just bare PDNP, the MIC is at 1 nanomolar. Okay, so what this demonstrates is PDNP, laser-induced photothermal heating, is indeed an effective strategy towards cell death or towards bacterial cell death. But we have a big problem here. So the big problem, even though it's an effective strategy, the big problem is that killing happens at 50 degrees. And if you recall in my, if you recall in the properties of an ideal antimicrobial, we actually don't want to kill healthy mammalian cell. We want our antimicrobial to be, we want our antimicrobial to also to not affect the healthy mammalian cell. And so, so basically the problem is that in addition to killing bacteria, it also kills the surrounding healthy cells. And in fact, this is a huge challenge in the in vivo killing of bacterial pathogens. And the challenge is to minimize collateral damage to surrounding healthy cells while at the same time specifically targeting the bacteria. So I was talking to other people about this, and they tell me it's like, oh, it's like, you know, trying to ask or demand the best from both worlds. But as I said, like that is research, people are looking for ways to mitigate this problem. And so I just want to mention here, as it will become relevant later on, it takes a temperature upwards of 42 degrees Celsius to kill a healthy human cell or to kill human cells, while it takes greater than 48 degrees Celsius to kill pathogens. Okay, so if we want to kill the bacteria, we shouldn't go above 42 if we don't want to affect as well the healthy human cells. So what did we do? So as I initially alluded to in my earlier slides, in addition to this laser-induced photothermal heating, we added lytic activities via antimicrobial peptide. And again, this is something that I already mentioned, but I just want to emphasize it here, that by combining the two, meaning laser, photothermal treatment, plus the dual function of antimicrobial peptide, we have the ability to, number one, target specifically the bacterial cell. And once that platform or antimicrobial is on top of the bacteria, it could start to kill it. So that's one way to kill it, the lytic activity of the peptide. And then if we hit it in addition, meaning we subject it to laser, there is an additional laser-induced photothermal heating. And that laser-induced photothermal heating is actually stimuli responsive. It is laser activated, near infrared, which is like, so we can penetrate into tissues in the body. Okay, so what I just said in few words, it means that we are able to target pathogens via multiple and coordinated biocidal mechanisms. Okay, so let's see if we are able to kill this bacteria. So here again is a plot of the bacterial growth as a function of nanoparticle concentration. The gray curve here is what you've seen in the previous slide for bare PDNP. So if you recall, the MIC for without the peptide, it's at 1 nanomolar. Now, when we confer added functionalities via the antimicrobial peptide, we see a slight improvement in the MIC. So from 1 nanomolar to let's say 0.6. So for the skeptics in the audience, you might say like, oh really? Like 1 from 0.6? Is that even an improvement? So just wait. This is actually not the highlight of our result. So if I bring up that temperature versus time plot that I showed you earlier, notice that 0.6 nanomolar dopamine, so let's say 0.5, that's close to it, the temperature only increases close to 40 degrees. Okay, so which means then, at this particular condition, meaning PDNP CWR11, we are able to kill bacteria, but killing happens at a much lower temperature than without the peptide. So instead of killing it close to 50 degrees, we are able to effect killing at a much lower temperature that are still compatible with a healthy mammalian cell. And so what this tells us, by the way, is we need this combination of multiple mechanisms. So not just from the laser-induced photothermal, but also for the lytic activities of the peptide. So you may want to ask, like, why is it possible, or how does it or what's the mechanism? So that's one thing that we were actually excited to look at. What's the mechanism for killing? So what I could tell you at this point is, imagine we have our antimicrobial platform with a peptide, which means that it could target the bacterial cell. So it means that it brings the antimicrobial closer to the bacteria surface. And once it reaches the bacteria surface, number one, it already starts its lytic activity. And on top of that, you shine laser to it, you will in a way, you will add more membrane damage. So for you to believe me, let me give you some evidence on, number one, specific targeting of the peptide, and number two, the membrane damage that this PDNP CWR11 actually induce onto the bacterial surface. And so to help us understand the mechanistic action of this antimicrobial is we turn to a favorite tool of mine. It's something that I've worked with as a grad student, even like in the postdoctoral labs that I've been to, this atomic force microscope, or AFM. So in the interest of time, I'm not gonna go through the details on how the AFM works, but then we use this nanoscale AFM tip, or we use this nanoscale technique for us to be able to image the surface topography of the bacteria, and also to measure the mechanical properties of the membrane. And that is important later on because we will use it as a measure of the degree of the damage that was done to the bacterial membrane. So first, let me show you the images that we obtained. So here is an E. coli and incubated with the pure or the naked or the bare PDNP, meaning no antimicrobial. It is actually interesting that for bare PDNP, most of the nanoparticles are found on the substrate and not much on the surface of the E. coli. So that's very interesting, especially when we compare it to E. coli that's incubated with the PDNP CWR11. So here you can see that these clusters of nanoparticles are mostly on the bacterial surface and rarely on the substrate. So this is a first one proof, a visual proof wherein the presence of the peptide in the nanoparticle indeed led to the more efficient binding. And once again, because of that, it results to what we call this like localized nanoheating. So when we apply laser later on, it just doesn't diffuse anywhere, but it gets localized onto the bacterial surface. Other people in the field calls it localized pasteurization. And that is the mechanism for this general hyperthermia, wherein you use laser or heat to kill a pathogen. So you rely on this heating, photothermal heating. Okay, so one thing that we also verified is our PDNP CWR11, is it still viable? Is it still active even when bound to the nanoparticle? And the reason why that's important to verify is people have shown, and it is actually fairly established, that once antimicrobial peptide is attached onto a surface, it actually loses its activity. Sometimes it could lose like 100-fold. But then here I'm showing you, these are for the PDNP in the presence of the peptide, with and without laser, the bacteria still, we can able to suppress the growth of the bacteria. So meaning the antimicrobial peptide retains its biocidal activity, or its biocidal activity is preserved even when bound to the nanoparticle. Okay, so this is an evidence for the efficient binding or the targeted binding. How about for the membrane damage? And so for the membrane damage, instead of looking at topography, we use AFM to measure the Young's modulus of the membrane. In other words, we use the stiffness. We look at the stiffness of the membrane and how it's being influenced by the action of the laser or the action of the antimicrobial peptide bound to the nanoparticle. So here to help you appreciate the plot that I put in here, I just want to note that higher Young's modulus means more resistant to deformation, meaning more destabilized membrane. So once again, we take it as a metric to quantify membrane damage. And so for the black box plot here, you can see this is our control E. coli, meaning no PDNP with peptide and no laser treatment. And the stiffness of the bacterial outer membrane, it's about 9 megapascal, plus or minus, I believe, like 4. In the presence of the laser, we see a decrease in the Young's modulus from 9 to about 5. So we are already starting to see that in the presence, this is just in the presence of the PDNP CWR11, so no laser. So again, it shows you the lytic action of the antimicrobial peptide bound to the PDNP. On top of that, if we induce it, if we use laser-induced photothermal heating, we get a more dramatic decrease in the Young's modulus. So you can see here from 9 to 5 to about 2 megapascal. So all these combined, it shows us that what we are observing if we combine this PTT, photothermal treatment therapy, and the action of the antimicrobial peptide, what we are observing is their synergistic effect. So we not only target the peptide specifically over mammalian cell, but we also are able to induce membrane damage, as indicated by a decrease in the stiffness of the outer membrane of the bacteria from 9 megapascal to 2 megapascal. So this is, I would say, as a result of the synergistic effects of combining multiple mechanisms. So you've seen this already. This is controlled E. coli in the presence of the nanoparticle with peptide. You see this now. If we add the laser in addition and we take the topography, you can actually see that we almost like kill the bacteria. So we induce so much membrane damage to it. So you see something what we call as like the ruffling of the membrane, which is also you will see if you just look at a free antimicrobial peptide acting on the bacterial surface. Okay, so I hope I was able to convince you that using this PDNP CWR11 antimicrobial platform, we are able to develop a nanomaterial-based antimicrobial that is biocompatible, exhibits multiple ways to kill the bacteria, can be externally triggered for its bactericidal activities, and also later on, it can be developed into a flexible platform. So once again, as I mentioned earlier, I could imagine putting antibiotics from, let's say, Doralyn's lab and seeing how it affects bacteria for one, and later on the biofilms. And speaking of biofilms, this is a very preliminary result. We tried looking into if this PDNP could actually penetrate into the biofilm. So here's a TEM image and of the cross-section of a biofilm, so not bacteria, cross-section of the biofilm. The gray bigger features you see here are bacterial cross-section, while the black dots are the bare nanoparticles. So here's the zoom-in image. And so without functionalizing the PDNP, we are able to penetrate about 13 microns already into the biofilm. I don't have data yet. I don't have data yet on PDNP with a peptide, but that is like something that we are working towards at this point. And lastly, for my last two slides, this is just quickly to tell you that a related experiment that the other postdoc is working on is developing tool to understand nanomechanics of this biofilm. So imagine I have this antimicrobial, I throw it onto my biofilm, or I treat my biofilm with it, and I wanted to understand what does it do to the stability of the biofilm, to the membrane of the biofilm. And so the way that we do it, we call it the quantitative imaging. It's a more, so in a way with a spatial, we look at data of the biofilm in a more spatial way, so more systematic, so that the goal is to be able to correlate biofilm structure to its mechanics, such as elasticity or maybe adhesive properties, if any. And also we could correlate the structure with the thickness of that EPS, or the biofilm matrix. And we do have data for that. This is also fairly new work, and literally last night I submitted the corrections for the proof. So this is the data by Christian. Here is your topography using QI imaging, and the corresponding layer thickness and stiffness. And I would want to show it because you can then imagine that we throw antimicrobials into it and we see how these properties change. So we can do, let's say, just mere like pure antibiotics, a cocktail of antibiotics, so as I showed to you today, and a nanomaterial-based platform, and we look at how it affects the properties of your biofilm or your bacteria. So in summary, I hope I was able to convince you that the combination of laser-induced photothermal heating along with lytic activities or membrane destabilizing activities of an antimicrobial peptide, that we can indeed have a multifunctional and stimuli responsive antimicrobial. And with that, I hope I did not go over time. I cannot see a time here, but I'd like to thank my group again, our collaborator from the Humboldt University in Berlin and the funding agency. And of course, to you for your attention. And once again, to Doralyn for this really great opportunity to share with you the work that we've been working here in Toronto, Canada. So I'd be happy to take any questions later.
Thank you very much, Ruby, for sharing the hot stuff, like fresh from your bench, right?
Yeah, so very fresh indeed.
These are exciting things that you are doing in your lab, and I'm very excited of like collaborating with you on this aspect. So
yeah,
thank you very much. So at this point, I will ask Mon if Ronald's file has been transferred, or can we view Ronald's file now? Can he share?
We'll try po with Sir Ron first before me po.
Okay, so.
Okay, I'm gonna try to share my screen. I'm trying to share it, but it wouldn't. For some reason, it's not sharing.
Plan B na po tayo, sir.
Okay, sure. First of all, okay.
Okay.
Is that okay now?
Do you see your presentation, sir?
Yeah, I see my presentation. So I'll just say next slide if
noted pa.
Yeah, we'll just wing it, I guess. Thank you, Doralyn, for the invitation, and also thank you, Dr. B, for filling in. So instead of talking about my overall work as a postdoc at Harvard School of Public Health, I thought I would just share a piece of work of mine regarding my overall work in finding the elusive asthma gene. So today, I would like to tell you a story about how a long-suspected asthma gene might actually be involved in the pathogenesis of asthma and how we can potentially target this gene into developing new asthma therapy. Next slide, please. So asthma is a chronic disease of the airways affecting hundreds of millions of people worldwide. Asthma not just robs the patients a good quality of life, asthma also kills. And asthma continues to be a major source of economic burden. For example, in the United States alone, asthma costs the US economy a staggering $60 billion annually. So really, asthma has become a global public health concern. So next slide, please. If you compare an asthmatic airway versus that of a normal airway, you will readily see the big differences between the two. An asthmatic airway, one of the striking differences is that an asthmatic airway has narrow lumen compared to that of a normal airway. And this structural change causes airflow limitation, which then leads to the difficulty of breathing and the shortness of breath that asthmatic patients experience during episodes of asthmatic attack. Now, lining the inside walls of the lumen is the airway epithelium, which is made up of many different types of cells, and which is actually the first line of defense of the airway against invading respiratory pathogens. Now, in asthma, there is evidence for epithelial denudation or epithelial damage, which is actually thought to result from programmed cell death. And this epithelial denudation, or epithelial damage is believed to be one of the earliest events in the pathogenesis of asthma, which leads to airway inflammation and the difficulty of breathing that the patients complain about. So despite the obvious significant progress we have made regarding our understanding of the pathophysiology of asthma, and despite the increasing prevalence and morbidity and economic burden of this disease, our understanding of the molecular basis for how a normal airway, could you, next slide, please? Our understanding of the molecular mechanisms for how a normal airway becomes an asthmatic airway remain incomplete. And this actually largely contributes to the fact that even until now, asthma remains an incurable disease. But if we actually knew the processes, then we should be able to convert an asthmatic airway back into a normal airway, or, probably halt the progression of the disease. Next slide, please. Next slide, please. So one of the reasons why asthma, or the molecular mechanisms of asthma, have been quite difficult to understand is because asthma is a classic complex disease, and like many other complex diseases, asthma is caused by mutations in many different environmental factors with many suspected asthma genes. This is in contrast with monogenic diseases whose etiology resides on mutations in a single gene. For example, the cystic fibrosis disease is caused by mutations in the CTFR gene. But if, next slide, please. But if complex diseases such as asthma are caused by perturbations in many genes in the human genome, so how do we search for genes that are potentially associated with the pathogenesis of a particular complex disease? And one approach is through genome-wide association studies, or simply called GWAS. Now, in GWAS, you actually search for genetic factors that are that might contribute for a particular complex disease. So essentially, you divide patients, or you divide populations into two. You have this is specific patients, for example, the asthmatic patients, and then you have the healthy controls, and then you isolate their DNAs, and then you scan their genome and search for genetic variants, otherwise known as single nucleotide polymorphisms, or SNPs. And once you have that information, you compare the relative frequencies of the SNPs from the diseased population relative to that of the healthy controls. And then you can plot these SNPs in a chart, for example, a Manhattan plot, which is going to be on the next slide. Next slide, please. So obviously, many GWAS studies have been performed on have been conducted on asthmatic populations. In fact, some of the earliest GWAS, some of the earliest seminal GWAS studies were conducted on asthma population to search for genes that might be involved in asthma susceptibility. And so here are just two of here's just one of the, actually two of the studies. Next slide, please. Okay, so here are just two of the Manhattan plots from the earliest, or from the early seminal GWAS studies. And by the way, a Manhattan plot is just a scatter plot. It's just so named Manhattan plot because it resembles the Manhattan skyline. So on the x-axis of this Manhattan plot are the chromosomes, and on the y-axis is the statistical significance, or the p-values. And the dots on this chart represent the individual SNPs across the human genomes. And as you can see from these two seminal asthma GWAS that were conducted almost a decade ago, you can see that there were many genes and many multiple loci that have been discovered to be associated with increased asthma susceptibility. But one locus, next slide, please. One locus stands out as being the most reproducible and the most consistent and the strongest asthma GWAS signals. And this resides in chromosome number 17, and it's called and it's referred to as the 17q21 locus. Now, again, the 17q21 locus has been the most reproducible and most consistent asthma GWAS signal. This has been replicated in many populations across many different asthma cohorts. And next slide, please. So if you zoom in on 17q21 locus, you will notice that this locus harbors 6 genes, the IKZF3, CPBP2, ORMDL3, PSMD3, GSDMA, and GSDMB. And you will also notice that there are many genes in the 17q21 locus, or there are many SNPs that reside in each of the genes of the 17q21 locus. Now, GWAS, because GWAS are just mainly, or they are for the most part association studies, we do not really know whether the association of the findings regarding the SNPs and the genes from GWAS are actually causal in nature, or whether their association with asthma is just purely correlation. So to resolve this dilemma, one would need to do a follow-up mechanistic or functional studies. And because such follow-up mechanistic or functional studies are lacking, the identities of the asthma gene still remains elusive. So part of one of my goals for this particular study is actually to identify which among these SNPs or these genes actually modify asthma risk, and if so, how do they modify asthma risk? So as you can see, there are many SNPs in each of the 6 genes in the 17q21 locus. And so if one would want to conduct a follow-up studies on the SNPs and the genes, how would he decide where to begin? So instead of studying these many SNPs and these 6 genes at the same time, that would require a lot of manpower, a lot of reagents, and a lot of time. So we instead prioritize only the coding variants, meaning these are the SNPs that are or that reside in the coding regions of the protein of the protein product of the genes. We also prioritize only the common coding variants, meaning these are the SNPs that occur with relatively high frequency in the human population. We actually set a threshold of minor allele frequency of greater than 5%. And then after populating these common coding variants, we then examine their association using using 2 existing GWAS cohorts, the GERA cohorts and the EVE consortium cohorts. And in the next slide, please, we're gonna see what we found. So actually we use this exome consortium browser or database to search for such common coding variants. And what we found was that there are actually 8 common coding variants in the 17q21 locus. And those reside in GSDMB, CPBP2, and GSDMA. And no coding variants reside in the other 3 genes of the 17q21 locus. So we actually excluded those for this particular study. So on the 1, 2, 3, 4, 5. I'm sorry, I don't have my the pointer here is not working on my side. But on the 6th column, that shows the coding change of the corresponding SNPs. So the first one, for example, RS2305480, that the change is actually a mutation in the proline at position 311 of the amino acid sequence being changed to serine. And the second one is RS11078928, which leads to a splicing change. And I'm going to talk about these two SNPs in detail later. So then, as I told you, we wanted to re-examine two existing cohorts, the Chera cohort and the Eve cohort. And next slide, please. And what we found is that at least the top two SNPs are nominally associated with lower or with decreased asthma risk in the Chera cohort. And we also replicated these results in the Eve cohort. But in the interest of time, I am not showing you that table, but it is in my first authored publication that came out 2 years ago. And next slide, please. So this information actually pointed at us to GSDMB gene for follow-up functional studies. So the next thing we asked is, so it seems like GSDMB is indeed one of the, or is the most important gene in the 17q21 locus as far as the cohort that we studied. So the next thing we asked is where in the relevant cells of the lung, meaning the cells that participate in the pathogenesis of asthma, is GSDMB expressed. So to answer that question, we did immunostaining of the human airway tissue section, and we found that GSDMB is highly expressed in airway epithelial cells. I do not have a pointer here, but it's shown in that green color, and its expression co-localizes with beta tubulin 4, which is a known marker of ciliated airway epithelial cells. And we are also able to replicate these findings with differentiated airway epithelial cells that are cultured in airway liquid interface. So we also did a bunch of qPCR analysis looking at the expression of GSDMB even in airway smooth muscle cells and in airway epithelial cells and in lung fibroblasts. And we consistently found that GSDMB is expressed in airway epithelial cells, but mostly in ciliated airway epithelial cells. So after knowing where GSDMB is expressed in the relevant cells of the airway, next slide, please. We then asked the question, what does this gene do? So at the time I was beginning to study this, I was studying the very first time I was, or during the initial stages of my endeavor to study Gasdermin B in the context of asthma, there was not really much known about the function of this gene, except that it belongs to a family of proteins known as the Gasdermins. And then the Gasdermins actually are Gasdermin A, B, C, D, and E. And each of the genes in the Gasdermin actually has human and rodent homologs or orthologs, except for, guess what, Gasdermin B. So that actually presents some challenges to us in later studies. But next slide, please. So also a few years back, we were very lucky because there were two seminal papers that came out in Nature that suggest, or that prove that Gasdermin D, one of the members of the Gasdermin family, in fact, one of the leader, quote unquote, or the leader of the Gasdermin family, is actually the long-awaited executioner of pyroptosis. And pyroptosis is a form of programmed cell death mediated by inflammatory caspases such as caspase 1 and 11, and is characterized by cell bursting and release of inflammatory molecules. So what happens during pyroptosis is that Gasdermin D is cleaved into an N-terminal fragment and a C-terminal fragment, where the N-terminal fragment migrates and and oligomerizes in the plasma membrane, where they form pores through which inflammatory molecules pass through, and this eventually leads to cell bursting and cell death. So these researchers, next slide please, were able to actually map out where the caspase-1 cleaves Gasdermin D. So they were able to map out that they were able to identify that caspase-1 cuts GSDMD in at an aspartic site in the linker of GSDMD protein. So because of the high sequence homology among the Gasdermin family, among the Gasdermin genes, next slide please, we hypothesize that Gasdermin B might actually be cut by caspase-1 as well. And so we look for aspartic acid sites in the region where it aligns with Gasdermin D, so close to that region. And then we found actually 2 aspartic sites, one in position 236, and then the other one is position 250. And so then what we did is we cloned the full-length Gasdermin D wild-type protein, wild-type cDNA, and then we mutated the aspartic sites at aspartic acid at 236 and 250 positions, or 250 positions into alanine. And next slide, please. And then we next slide, please. We transfected this into 2932 cells, and we performed a very simple traditional biochemical caspase-1 activity, enzyme activity assay, where once we have the lysate of the transfected cells, we mix it with caspase-1, very simple assay. And what we found are actually 2 things. First, caspase-1 cuts the wild-type GSDMB as shown by a low molecular band there, somewhere between 20 and 15 kDa, right? There's second column in the Western blot, if you can see. I don't have a pointer, I'm sorry. And the second finding is that caspase-1's cutting site on the Gasdermin B is actually in the position at the aspartic site, because with the mutant, when we mutated aspartic acid into alanine, that cleavage column number 4 is now abolished. So next slide, please. So we actually replicated this finding with a co-transfection of the different mutants with caspase-1, and again we confirmed that indeed caspase-1 cuts GSDMD, and it cuts the protein at the D236 position. So next slide, please. So next slide, please. So this is what's actually happening. Yes, so if Gasdermin B is cut into an N-terminal fragment, does this also, or does the N-terminal fragment also cause pyroptosis or toxicity? So we can easily measure toxicity by pyroptosis through an assay called LDH release assay. Very simple assay. It's just a colorimetric assay measuring the release of the lactate dehydrogenase enzyme. And what we found is that transfection of N-terminal fragment here induces massive pyroptosis in the transfected cells, but neither the full length nor the C-terminal fragment of GSDMB causes such toxicity. And on the second panel just shows you the representative images where the N-terminal, where I'm showing the N-terminal transfected cells are rounding up. That's in the, here in the third image. The cells are rounding up, and this is very characteristic of pyroptosis. They are actually undergoing cell death, and then they are detaching from the bottom of the plate. And as you can see, the EGFP control, the full length, or the C-terminal fragment, they did not cause these morphological changes. This cytotoxicity induced by the GSDMB N-terminal fragment is so fast. This is massive. Within 6 to 12 hours of transfection, the cells would just start to completely die, and that would, would start to die, and the entire process would almost be complete in like 18 hours or so. On the third panel is the caspase-1 co-transfection with the wild type as well as with the mutants. And again, wild type causes toxicity because it can be cut by caspase-1, but the D236A mutant, because it wouldn't be cut by caspase-1, so it wouldn't show any toxicity at all. So next slide, please. So you may be wondering, how does this relate to the two common coding variants that I talked about, or that I had talked about earlier? And now I wanted to, if you recall, the two coding variants are RS2305480, which leads to a coding change or mutation of proline to serine at position 311S. And then the other one is RS11078928, whose T to C change destroys a splicing acceptor site. So this is a splicing variant. And next slide, please. So what happens is that this second variant leads to exon skipping of the entire exon 6. And the exon 6 actually becomes deleted. Next slide, please. And so the resulting variant of the second, the resulting protein product of the second variant, the splicing variant, is actually 403 amino acids less than the wild type. And the first variant, because I forgot to emphasize, the first variant, the RS230548, which is P2311S mutation, the mutation actually resides in the C-terminal fragment. So the splicing variant, RS11078928, actually again, it results into a protein that is 13 amino acids less than the 400, less than the wild type, but it does retain the caspase-1 cutting site. And next slide, please. So we look for homozygous allele, homozygous minor allele, those donors with the homozygous minor allele with the variant, and we perform RT-PCR, or reverse transcription PCR, and as well as Western blot analysis to check whether indeed the splicing variant would result in the deletion of the 13 amino acids. And indeed, here I'm showing, at least in the RT-PCR, that the splicing variant RS11078928 deletes the 13 amino acids from the GSDMB protein. Next slide, please. So then we clone the N-terminal fragment of that particular variant. Again, that particular variant retains the caspase-1 cutting site, so it would still be cut, but then it has a deletion of 13 amino acids in the N-terminal fragment, so it has shorter N-terminal fragment. And so we transfected this N-terminal fragment from the variant to the cells. And what we found is that this particular N-terminal fragment does not or fails to induce pyroptosis. This is actually the variant, the shortened GSDMB by the variant. So next slide, please. So we also perform co-transfection of the full-length wild type and the P311S, and then the 403 GSDMB, that is the splicing variant. Next slide, please. And we showed that in the 403 GSDMB, the full length, even if it can be still cleaved by caspase-1, it doesn't cause any cytotoxicity as compared to the 416 and then the other variant, because the other variant can still be cleaved, but the mutation is in the C-terminal fragment. And next slide, please. So we were also able to replicate these findings by transfecting, by doing the same experiments in two other airway epithelial cells, BIS2B and 16HB, and we are able to essentially confirm, to essentially observe the same results. So this suggests that the shortened GSDMB by the RS11078928 variant, or SNP, fails to induce pyroptosis. It appears that the 13 amino acids that are deleted because of the exon skipping are actually important in the pyroptosis activity of the N-terminal fragment of the GSDMB gene. Next slide, please. So I'm just gonna do a quick summary, and this is how we visualize the role for the potential role or functional role of GSDMB in pyroptosis and then in asthma. So in ciliated airway epithelial cells, GSDMB protein is produced. Next slide, please. Where in the presence of caspase-1, next slide, please. In the presence of caspase-1, this can be cleaved into two N-terminal, into two fragments: the N-terminal fragment and then the C-terminal fragment, where the N-terminal fragment migrates migrates and oligomerizes on the cell membrane. Next slide, please. And the N-terminal fragment forms pores. Next slide. They form pores through which cytoplasmic contents, such as inflammatory cytokines, can pass through, and this eventually leads to pyroptosis. And this is what we think is happening during asthma attack, or after the during episodes of asthma attack. Next slide, please. There you go. Next. Yes, go on, please. So that's the pore. Go on, please. Yes, just continue. So that's the pores that I am visualizing in my mind. So yes, please go ahead, go on. There you go. Yes, and please next. Then, if you have the variant, for those people who have the variant, remember this is a variant that is associated with decreased asthma risk. So you're going to have exon skipping, and that would lead to a sort of a different form, or a shorter form of GSDMB. And then in the presence of caspase-1, that would still be cleaved, as you can see here. But then the N-terminal fragment now has a deletion in the with deletion in the N-terminal fragment by 13 amino acids, and somehow that doesn't form pores in the cell membrane. And that doesn't somehow lead to, next slide, please, pyroptosis. So there is no pyroptosis. I'm waiting for the next slide. Yes. Okay. So where do I go from here? So as you can see, there are many causes of asthma. As I told you in the very beginning of the talk, asthma is caused by complex interactions of genes with environmental factors. And so what we want to do for my follow, and in fact, this is what I'm doing right now in the laboratory, is we're trying to prove trying to identify which among the asthma triggers, such as rhinoviruses or pollen or house dust mites and chemicals or tobacco smoke, which among these actually lead to caspase-1 generation, or which among these can potentially cut the GSDMB protein itself. And so then this is how we're going to tie everything with in the context of asthma. And next slide, please. So that is actually where the direction where I am leading right now. Next, please. Next slide, please. So just very briefly, next slide, please. Okay, so just very briefly, so I was able to identify one asthma trigger. So I'm just not going to disclose that particular trigger because we are in the process of performing more experiments. The asthma trigger X actually cleaves GSDMB protein. And so on the left is just the dose response, and on the right is just the time course assay. And on the next slide, please. On the next slide, I was able to, or we found one FDA approved drug that actually can block the the cleaved GSDMB induced pyroptosis. Next slide, please. And next slide, please. Anyway, so so on the next slide is the FDA approved drug Z that we were able to show to block the cleave induced the cleave GSDMB or the N-terminal fragment induced cytotoxicity. And what we're hoping to do next is to actually perform a high throughput small molecule inhibitors screening to identify which other drugs actually or could prevent the cleavage of GSDMB or prevent the toxicity induced by the N-terminal fragment. So if that's actually my second to the last slide, and so I'm just gonna go ahead with the acknowledgement. So as you can see, this is a very collaborative, or as if you realize, this is a very collaborative work. I am basically a molecular biologist, and I collaborate a lot with molecular epidemiologists and computational biologists, and even physicists and mechanical engineers for this project, and even for a lot of my projects in the laboratory. So this would not have been possible without their help. So on the last slide is actually my acknowledgement. So I would just like to thank them and thank you, and I'm sorry and my apologies about the glitch, and I think I am ready to take your questions.
So thank you very much, Ron, for sharing your work on asthma, and we really appreciate your time and effort for sharing this to us, because it's already 11 p.m. in the East Coast, and and then
no worries.
Play for this opportunity to share your work is something that we really appreciate. And so at this point, we will have an open forum. And so I'm going to share some of the questions here. So first, we are going to have a question for Ruby. Ruby, are you there?
Yeah, I'm here. Yeah.
So we have actually two questions. So the first question is, the arginine and tryptophan residues in CWR11 peptides suggest possible good interaction with the membrane phospholipid head groups and insertion of the side chain in the phospholipid tails in the bilayer mammalian cell and possibly in bacteria. So the first question is the peptide conformation ordered or random? And second, does this peptide spontaneously traverse the membrane or trigger endocytosis or sense membrane curvature? And the second one, how do you envision the use of nanoparticle-based antimicrobials in clinical, animal, or agricultural settings? How will you ensure that the safety of using dopamine-based antimicrobial nanoparticles, and if these are biodegradable by metabolizing enzymes in the body? So go ahead.
Yeah, so first, thank you for the question. So the number one, I recognize this is from Janelle. So that's a good question, Janelle, that I did not mention about anything about the structure of the peptide. So this is a short peptide. So your question is the peptide conformation ordered or random? So it's not random. In water, it's actually a combination of alpha helix and beta sheets. The interesting thing is that if you put it, so the authors in the paper, they put it in an SDS that is membrane mimicking. And by doing that, the order of the structure of the peptide actually changed from a mixed helix and beta sheet to more alpha helical. And then when they throw the CWR11 into PBS, they again see a alpha helix structure. So I would say in the more relevant environment, they turn into alpha helix. As to your question of it spontaneously traversed the membrane, it does. There is some evidence to that that's been showed also by the group. And they hypothesize that the reason it does so is because your arginine residues, it forms hydrogen bonds with the surrounding water molecule. And in addition to that, it also formed because I mentioned that peptide charge is +6. And so it forms a cation pi interaction with the tryptophan. And so the authors hypothesize that the combination of these multiple hydrogen bonds with water molecules for one, and also this cation pi interaction with tryptophan, it actually makes the insertion into the membrane very favorably, it's energetically favorable. So and on top of that, they have some evidence that actually shows that they were able to traverse all the way to the inner membrane. As to your other interesting questions, endocytosis and membrane curvature, I actually don't know. They haven't demonstrated it yet. So at least in the people who've used it, they did not use it for mammalian cells yet. So no evidence on endocytosis or that it can able to sense endocytosis or membrane curvature. So hopefully that answers your questions, but then feel free to email me offline, and I'll be happy to chat with you. So the second question, so this is interesting. How do you envision the use of the nanoparticle in clinical, animal, or agricultural setting? So by the way, I would like to mention that I get that question every time. And one funny question I got is like, are you gonna photothermal the entire person? Because I was like selling it via laser-induced photothermal heating, right? So, so the way we envision it in terms of application, it can be used to treat localized infections. So it's either wound or like any localized infection. I did not show a good plot that shows you the NIR laser. It's 808 nanometer. So 808 nanometer, it means that it could penetrate into tissues like without causing any damage. So meaning we could do this, we could use this NIR laser photothermal induced heating onto localized sites of infection. On top of that, it can also be used, let's say, to treat medical implants or medical devices. That way, you can use that for that. And the work that I showed you is from the Nesha, the one graduate student in the group. She is actually focusing similar project, but then the application is towards dentistry. So in teeth. So you can easily imagine it's not very difficult to do photothermal treatment to your teeth. So that's how we envision on the use of the microbial. How will you ensure the safety? So I mentioned at the start that dopamine is a neurotransmitter. So it's already found inside the body. So we're not actually worried about the cytotoxicity of dopamine itself. But then I did not show in the interest of time in my slide, but we've done some hemolytic studies of this PDNP CWR11. So I don't know if I could quickly share with you here. It's just right here. I have it as a backup slide. Oh yeah, perfect. So here's the hemolytic study. So you can see this gray here is just the bare nanoparticle. The green one is just the free peptide, and the red one is our PDNP with the peptide bound. So it's actually not, it's very close to without anything. So and in the presence of the free peptide, it could reach up to 60% hemolytic activity, while our PDNP CWR11 is less than 20%. So hopefully that answers your question. I don't know if there's a third one, but I think I did address it.
Okay, thank you very much, Ruby. Thank you very much for answering those questions. And now we are going to have a question for Ronald. So Ronald, you have questions here. First one, among the caspases family, is caspase-1 the only trigger for pyroptosis through GDSMB cleavage? Since caspase-1 is also involved in apoptosis, does pyroptosis simultaneously occur with apoptosis? Second, in the study portrayed by Gasdermin B as an asthma-related gene, is the mechanism of this gene exclusive only to asthma, or there are other respiratory-related conditions that this gene could contribute for their pathogenesis? And third, what are the difficulties you encountered while doing genome editing, and how did you overcome these difficulties? So Ron.
Thank you. Okay, thank you for the questions. Those are very good questions. So for the first one, caspase-1 is at least, so Gasdermin D can be cleaved, Gasdermin D, not B. The original paper suggested that it can be cleaved by caspase-1, caspase-11, and some also said that caspase-4 and 5 and many other caspases. But on our end, we were able to only show caspase-1 to be the caspase that could cleave Gasdermin B. We tried to do caspase-4. I didn't show the data, but caspase-4 would not cleave Gasdermin B. So since caspase-1 is also involved in apoptosis, does pyroptosis simultaneously occur with apoptosis? Caspase-1 is mainly for pyroptosis, but there were also reports that it does affect or it does induce apoptosis. And my answer to that second question of the first question, second part of the first question is, I am not actually really sure, but because I haven't really measured apoptosis simultaneously with pyroptosis, but that's something that is worthwhile doing. And the second question, this study portrayed Gasdermin B as an asthma-related gene. Is the mechanism for this gene exclusive only to asthma? Or are there respiratory-related conditions that this gene could contribute for their pathogenesis? So Gasdermin B is also one of the genes identified by GWAS to be associated with inflammatory bowel disease. So it's potentially something that I think probably the process that happens in inflammatory bowel disease in the context of Gasdermin B is the same as that's what's happening in asthma. But I am not an expert in that particular disease. So I could only guess that it's going to be the same mechanism. With regards to whether it's might be involved in other respiratory related conditions, I actually I've been thinking about that. And if ever I would have my own lab in the future, then probably I would dig deeper into that. I was thinking at one point that it might be a mechanism for the how COVID or how SARS-CoV-2 cause or lead to exacerbation or like lead to the inducing the toxicity in the respiratory system. And for the third question, what were the difficulties you encountered while doing genome editing, and how did you overcome those difficulties? So I haven't really showed any, I didn't really show any data that had something to do with genome editing with regards to the variant splicing, but we actually attempted those. I did a lot of I did genome editing by CRISPR through by CRISPR-Cas9 with the long template. But there are two things here. One is that the GSDMB is actually expressed only in airway epithelial, mostly in airway epithelial cells. And I cannot just, for example, I cannot just get a 293 T cells, which are very good in doing genome editing, because they would not really, they do not express GSDMB. So I have to go to the differentiated airway epithelial cells. So that presents another challenge, because that's kind of like a tissue already, because we can only study, GSDMB is only expressed in differentiated airway epithelial cells. Second is I cannot just get mouse cells, because it is absent, GSDMB is absent in mice. So those are some of the difficulties. Genome editing should be not very difficult. I've done genome editing in some cells, but in this particular case, it was difficult just because because of the expression of GSDMB only in certain tissues. Thank you.
Okay, thank you, Ron, for answering those questions, and thank you very much, Ruby and Ron, for sharing your time again and expertise. We really appreciate that. So let's move on with the last and the third topic. This is drug diagnosis and drug delivery. Our next presenter is Dr. Bernadette Marques Nostra. She is a PhilAm and currently an assistant professor in the PET Center and the Department of Radiology and Biomedical Imaging at Yale University. She received her bachelor's degree in biochemistry at California State University in Fullerton and her PhD in chemistry at the University of California, Davis. She trained as a postdoc fellow at the Washington University and St. Louis, and she now leads the Laboratory of Cancer Imaging and Therapeutics at Yale that focuses on developing nuclear imaging agents to predict or monitor response to cancer therapy. So ladies and gentlemen, I would like to present to you Dr. Bernadette Marques Nostra for her talk on the PET-based companion diagnostics to predict response to targeted therapy. So Bernadette, the floor is yours.
Thank you so much, Doralyn. Can you hear me okay?
Yes.
Great. And also thank you very much to Janelle, and hello to Joan. It's been a very long time since I've seen you both. So tonight, I'm still on August 5th. Tonight, I'd like to talk about and focus on my talk on PET imaging and how we can use it to predict response to targeted therapy. So mostly I'm going to talk about antibodies because they're very versatile. They can inhibit various signaling pathways, deliver cytotoxic payload if you conjugate cytotoxic drugs to it. Recently, several FDA approved antibodies have been used to activate the immune system for immune therapy. And finally, you can modify antibodies with radioisotopes that emit either imaging or therapeutic properties. So currently, patients for antibody treatments are selected based on what's called companion diagnostics. By definition, the FDA calls this a medical device or an in vitro device that provides information for the safety and effective use of a corresponding drug or biological product. And an example of a companion diagnostic is immunohistochemistry, or IHC. So normally patients would get a biopsy of their tumor, and then the tissue would be processed and stained for the antigen that would tell you how much of the antigen is present in that tumor tissue that would allow that patient to be eligible for that antibody treatment. However, there are some limitations to tissue-based analysis in general, in that a biopsy is very small, and often patients with metastatic disease have heterogeneous expression of the antigen of interest. So here is an example of heterogeneity within a tumor, where some parts of the tumor are scored very highly. In this case, this is a stain for the human epidermal growth factor receptor 2, for which an antibody called trastuzumab is used for treatment of this disease, while the rest of the tumor is scored very low. So the goal of my lab is to develop new companion diagnostic imaging agents based on PET imaging. And the idea here is not to look at a small tissue, but to look at the expression of the antigen throughout the whole body. So where ideally patients would be injected with the radio-labeled antibody, and those who have high uptake in their tumor lesions would be eligible for the corresponding antibody therapy. And those who have low uptake in their tumor lesions should get an alternative treatment. So how does PET work? Well, first, you have to produce the radioisotope, and this radioisotope has to have positron emitting properties. So for example, yttrium-89 is produced in a biomedical cyclotron, and yttrium-80, you bombard yttrium-89 to produce the zirconium-89 radioisotope, which then emits positrons for PET imaging. Then you would incorporate the zirconium into the antibody by radio labeling it, and I'll go into more detail in a few slides. And then it would be injected into a mouse-bearing tumor or into a patient. And then that mouse or that patient would be scanned in the PET scanner. What's happening in the PET scanner is that as the radioisotope decays, it emits the positron. The positron then annihilates with an electron inside the body, creating two opposite 511 keV photons that would be detected simultaneously around this ring of detectors inside the PET scanner. And so then the image would get processed, and then you would be able to visualize the image where the hot spot here, the red spot, would correspond to the accumulation of that radioisotope. In this case, this is the tumor. So for the advantages of PET-based companion diagnostics is that you are able to visualize the delivery of the antibody to the tumor and also look at its distribution throughout the whole body. So here is a patient with metastatic breast cancer. Again, I mentioned her too. And she was injected with zirconium-89 trastuzumab, where you can see distribution of the radio-labeled antibody in the lesion on the shoulder, on the hip, and also on the thigh. And you see clearance of the antibody in the liver. The other uptake that you see here are in the blood, because antibodies circulate in the bloodstream for a very long time. So with this technology, you can assess the antigen expression throughout the whole body non-invasively. And this will allow you to potentially select patients who are most likely to benefit from these antibody therapies, and clinicians will be able to plan or adjust their treatments. Zirconium-89, as I mentioned, is a transition metal. It has a half-life of about 3.3 days. As you can see, here is a decay scheme for the zirconium-89 to the stable yttrium-89. The decay of the positron, 23% of the time, is what allows us to see this isotope by PET imaging. So as I mentioned earlier, it's produced in a biomedical cyclotron by bombarding the yttrium-89 metal with protons. It would emit a neutron, and then it would create the zirconium-89 isotope. Zirconium-89 is nice because it does not have any known biological function in the body, and so it cannot be metabolized the way other radioisotopes are. For example, radioiodine is transported to to your thyroid. So if you were to label antibodies or nanoparticles or cells with zirconium-89, it would deposit where these targeted vectors deliver the radioisotope, and it would just stay there, which would allow you to get high resolution images. And so, like I said, zirconium-89 has a half-life of 3 days, which pairs well with the long circulation half-life of antibodies in the bloodstream. So this allows us to image at extended imaging time points. So here you can see at 2 days, you can barely see the uptake of the zirconium antibody into the tumor. However, waiting at 4 days, you can clearly see the resolution of the uptake of this radio tracer into the tumor, and know that that tumor is HER2 positive. So the project that I'm working on currently is imaging a receptor called glycoprotein non-metastatic melanoma B, or GPNMB, which was a promising target for triple-negative breast cancer. It won't focus too much on the biology of this receptor for the sake of time, but just know that high expression of GPNMB in patients is associated with lower overall survival. This is a good target for antibodies because in disease and tumor cells, it's expressed on the surface, while in most normal cells, it's expressed intracellularly. So marking GPNMB, very good selective marker for the cancer cell. Previously, there was an antibody drug conjugate called glembatumumab vedotin, or Glemba for short. It is an antibody that's linked to a valine citrulline linker and a monomethyl auristatin E drug. So this is a microtubulin inhibitor. When the antibody drug conjugate, or ADC, binds to GPNMB on the surface of the tumor, it's then internalized into the cell, and then gets the linker gets cleaved by specific proteases in the cell and delivers the drug to elicit an apoptosis and kill the tumor cell. However, recently the phase 2b clinical trial for this antibody drug conjugate failed. In this trial, patients with triple-negative breast cancer responded initially. However, they developed resistance, and even though compared with the investigator choice chemotherapy, there was more tumor shrinkage with the antibody drug conjugate. This resistance to this drug rendered or ended the clinical trial for this ADC. However, we think that GPNMB is still a viable target. May future work would be to look at new drugs to conjugate to this antibody. So my research focuses on PET imaging, and so we asked whether PET imaging of the zirconium labeled antibody, so the antibody without the drug, can predict response to the antibody drug conjugate therapy. So to create a PET imaging agent using antibodies with zirconium-89, the antibody first needs to be conjugated to its chelate. In this case, this is a desferoxamine chelate. Excuse me. Which is linked via the lysine residues of the antibody, and then radiolabeled at neutral pH with zirconium-89. So then we have to characterize the radiolabeled antibody to determine whether we've affected its antigen binding properties. And so by conducting competitive binding assays, we know that we haven't negatively affected the binding of the conjugated antibody. And then we looked at specificity and various cell lines that express different levels of GPNMB, where we see highest uptake of the zirconium antibody in the cell line with the highest expression. And so 231 MDA-MB468 here is moderate, 231 is negative. SKML2 is a melanoma cell line control. And so when we block when we treat the cells with an excess amount of unlabeled antibody, we can block the uptake of the zirconium labeled antibody to show that the binding is specific to GPNMB in these cell lines. So since we know that it's important for the antibody drug conjugate to internalize in the cell, we also characterize the rate of cellular internalization, where we found that the ADC internalizes at a fast rate within 30 minutes. So then after we do our in vitro studies, we have to then move into our in vivo experiment. So we establish patient or cell-derived xenografts. Cell-derived xenografts are cell lines that are commercially available that we can inject in subcutaneously on the of the mouse, whereas patient-derived xenografts are derived from patient tumors. So from that tumor biopsy, it's then propagated in a very immunocompromised mice so that the mouse doesn't reject the tumor, and then it keeps getting propagated by injecting and growing the tumor into more mice. And that's how we generate patient-derived xenografts. So once we form the tumors on the shoulder, we then inject our zirconium antibody, and after some time, we wait for the antibody to distribute and accumulate in the tumor, and then clear from the bloodstream before we image with PET. And after we do our imaging, we then treat the mice with the antibody drug conjugate. Then we measure the tumor size for 60 days. So here is an example of the pharmacokinetics of the zirconium antibody. As you can see, at 2 hours, it gets all over the place. And then at 1 day, 4 days, and 7 days, it clears from the bloodstream and through the liver. And you see an enhanced uptake in the tumor due to binding to GPNMB positive tumor cells. As a control, this is the isotype control, where you see the same distribution at early time points, and at later time points at 7 days, there is no uptake in this tumor here. So then we looked at 4 different models of triple negative breast cancer. The MDA-MBs, 231 and 468, are the cell-derived. This is negative for GPNMB. This is moderate for GPNMB. MK33 is a patient-derived xenograft. So this tumor is from a patient with primary breast cancer. 15017 is also another patient-derived xenograft that was isolated from brain metastasis of TNBC. So as you can see, you get different levels of uptake of the zirconium antibody into these very different models with varying expression of GPNMB. And this is how we quantify PET imaging by using standard uptake values, which is the concentration or the percentage of the radioactivity in a given region of interest and normalized to the body weight of the animal. And so then after these same animals were treated with the antibody drug conjugate, we measure the tumor size, and those tumor models with GPNMB positivity all responded to the antibody drug conjugate, while the 231 GPNMB negative tumor model progressed. And then we then correlated the percentage of the change in tumor volume from baseline with the SUV from imaging. And then we were able to show a correlation, a good correlation at 3 weeks post-treatment. So sorry. So, and with that, I'd like to thank Supam. This is mainly his project. He's a research assistant, and my postdoc, Alessandra, for helping with the radiochemistry. Zichi Lee is a new postdoc in my lab. And also my collaborators, Celldex Therapeutics, for providing the antibodies, and Sandeep and Al Bothwell for providing the patient-derived tissues. I'll take questions if there are any.
Okay, so thank you very much, Bernadette, for sharing your work with us. We will wait until Joan will present her work, and then after that we will do the question and answer. So now we have our last but not the least speaker. So we have Dr. Joan Schillinger. She is actually a native of the Gupan City, Pangasinan. She earned her bachelor's degree in biochemistry at University of the Philippines, Manila, and she pursued her PhD in organic chemistry at the University of California, Davis, working with Professor Jacqueline Gervais-Haig, where she designed and synthesized small molecule linkers and multivalent peptides for cancer therapy and HIV vaccine. Following her doctoral studies, Joan conducted her postdoctoral research at the University of Washington in Seattle under the supervision of Professor Susie Poon, and her postdoctoral work focused on the synthesis and evaluation of peptide-based polymers as gene delivery agents. Joan then joined the Department of Chemistry and Biochemistry at the University of San Diego, California in 2014, where she is currently an associate professor and working with undergraduate students to synthesize and evaluate derivatives and peptides and polymers of different biomedical applications and in origins of life. So ladies and gentlemen, I would like to present to you Dr. Joan Schillinger for her talk on RAF polymerization of cation polymers, synthesis and applications. So Joan, the platform is yours.
All right, thank you so much, Ate Doralyn. Okay, magandang umaga, tanghali po sa lahat dyan sa Pilipinas. Thank you very much, Ate Doralyn, for that kind introduction. It's great to see everyone, Kuya Janelle and Bernadette. It's great to see you. It's been a long time, I agree. It's amazing to be part of this seminar series, and it's just a pleasure to share with you our work at University of San Diego. So please allow me to begin just to share with you a little bit about our campus and my research lab. So University of San Diego is a medium-sized private Catholic university located at Central San Diego. So we're actually just south of La Jolla, where UCSD is located. So we are considered a primarily undergraduate institution. So all the work that I will be showing to you with my independent career at USD are all done by these amazing undergraduate students. So working with them has been really amazing, and it's definitely one of the highlights of my career. So my lab is an organic chemistry lab, and we do have a broad set of projects ranging from synthesizing small molecules, peptides, and polymers with different applications. So one project that we're working on is a collaboration with the professor at UCSD, where we are investigating peptides and their roles in the origins of life. Another project that we're working on is with a collaboration with a colleague of mine at USD, where we're developing this starch-based hybrid biomaterials for delivery. And the work that I will be focusing on today, and the work that I'll be sharing with you today, involves the preparation of these cationic polymers and the methodology that we developed in our lab. So the way we got started in this project with these cationic polymers is through my work as a postdoc in Susie Pan's lab at University of Washington. So just let me share with you a little bit about some of my work to give context of what we're doing at USD. So when I joined Susie Pan's lab, my main project was to develop this multi-component peptide-based delivery systems that targets neurological diseases such as Alzheimer's disease. And as we know, there's just a lot of people affected by it, and there's still limited treatment options. So the way gene therapy works is that it involves the introduction of a therapeutic gene into a host cell to target different diseases or disease phenotype. And the way to do that is that this therapeutic nucleic acid gets into the cell, gets delivered into the nucleus, and then translated into a functional or healthy protein. In some forms, it involves the delivery of a small interfering RNA that would induce gene knockdown. So in order to do this, there needs to be some kind of a delivery vector to introduce the therapeutic gene. So there's the viral and then the non-viral vectors. And we are interested in developing cationic polymers as non-viral vectors for gene delivery. So how these cationic polymers work is that the cationic component would electrostatically interact with the negative phosphate backbone of the plasmid DNA to form these nanoparticle complexes, also known as polyplex. One thing I should note that it's just a hard field. There's just a lot of challenges, problems that is involved in a polyplex mediated gene delivery. For example, the condensation, the cellular uptake, escape to the endosome lysosomal network. So in order to address these barriers or issues, our goal is to develop this multi-component peptide-based polymer system. Dr. Ruby Sulian gave a wonderful talk about the importance of developing multi-component materials for effective and functional materials. So in order to achieve our goals, we're utilizing peptides as our bioactive component. And as we know, a lot of people are interested here with peptides. They are very attractive molecules because of their biodegradability, specificity, and chemical diversity. So you can fine tune their structure to achieve a certain activity or function. So we wanted our polymers to be degradable or biodegradable in vitro and in vivo. So we're also incorporating a small molecule hydrophilic monomer known as hydroxypropyl metacrylamide. I'll just call it HPMA from now on. We wanted these polymers to be biodegradable for reduced toxicity. And also we wanted to utilize the versatile method of making this polymer so that we can have control in terms of incorporating these bioactive components. So there are a lot of polymerization methods that are available out there, but we chose to pursue the reversible addition, fragmentation, chain transfer, or I'll just call it RAF from now on. We chose to pursue RAF polymerization because it's easy, it's very versatile in terms of the use of different monomers, and then it produces polymers with this narrow molecular weight range or dispersities close to 1 or less than 1.2. So what this means is that the polymer chain lengths are closely in uniform lengths, so the dispersity are closer to 1. This polymer also, like I mentioned, gives control. I'm sorry about that. Gives control in terms of being able to incorporate different monomers in varying amounts and allows us to give polymers with different structures. And also this polymerization method can be done under mild aqueous conditions. So just a little bit of organic chemistry here. I'm an organic chemist, but basically how RAFT works. So RAFT is considered a living free radical polymerization. So it actually follows some similar steps in a free radical process that we learned in undergraduate organic chemistry. So you have the initiation steps, the propagation steps, and then the termination steps. Now, do take note that once the polymers undergo termination, you now have dead polymers that can no longer elongate. So how RAFT is unique compared to a conventional free radical polymerization is the presence of this chain transfer agent right here. So what it does is it facilitates some additional steps highlighted here. So the chain transfer agent reacts to the propagating free radical chain and then goes into this rapid equilibrium between its active and dormant state. So what it basically does, it caps the propagating free radical in order to prevent or minimize the termination steps. So we demonstrated, or we did our initial studies using RAF polymerization, using HPMA, and co-polymerize it with a peptide monomer, oligo-L-lysine here. And we have synthesized a series of copolymers with varying peptide lengths, different peptide composition, and different polymer molecular weight. So DP here refers to degree of polymerization, which describes the average number of monomer per monomer repeat unit along the polymer backbone chain. So that's basically your X and Y right there. So in our transfection studies, we have identified that a polymer containing oligolysine with 10 lysine and polymer containing 20% peptide and a polymer with a DP of 150, which is about 70 kilodalton in size, are the ideal or optimal delivery vectors for our transfection studies. So we utilize these parameters to synthesize our parent polymer here and to further develop our multi-component systems. So as mentioned earlier, one of the barriers for gene delivery is the escape in the endosomal lysosomal network. So once this polyplexes gets internalized, they can get entrapped in these vesicles and eventually get degraded. So the goal is to facilitate endosomal escape to ensure the cytosolic delivery of our therapeutics. So the endosomal escape peptide ligand that I investigated is melittin, which is a 26-mer membrane lytic peptide derived from the honeybee venom. So it its amphipathic and helical feature allows for it to insert into the lipid membrane and induce destabilization and pore formation. So just briefly on the synthesis of our melittin-based polymers. So using RAF polymerization, we made the first block using HPMA and this pyridyl disulfide monomer, which will be used later on for melittin conjugation. Once we've made this intermediate polymer, we then extended the chain by copolymerizing it with more HPMA and then the K10 oligolysine monomer to form our polymer 2 intermediate. So then we conjugate the cysteine modified melittin to this pyridyl disulfide moiety via disulfide exchange, and we monitor the conjugation efficiency using UV-Vis. And based on our studies, we were able to incorporate an average of 5 to 6 melittin per polymer. So we did a lot of structural characterization as well as in vitro transfection, but in the interest of time, I'm just gonna highlight the in vivo result of our studies. So the way we do or evaluate our delivery work or our transfection studies as well is using a reporter gene that expresses firefly luciferase. So with the addition of a reagent that contains the substrate for luciferase, you can measure the amount of light emitted, and it actually increases linearly with the amount of luciferase protein produced. So previously with our parent polymer that is melittin free, we do not see any gene expression at all. But after these studies, we were so excited to see gene expression or luciferase expression with the use of melittin diblock. So the way we do this, I should mention, is that we formulate our polyplexes by mixing this polymers with the luciferase plasmid, and then we inject it directly to the brain of the mice at the right lateral ventricle. And really, to our excitement, we saw a statistically significant increase in gene expression as the melittin concentration increases. So that's really encouraging. So we did a lot of we developed, synthesized, and evaluated a lot of different peptide-based polymers for gene delivery with different bioactive components. We also, for example, we utilized a shortened HIV glycoprotein as another endosomal escape ligand, a TET1 peptide as targeting ligand, and we also evaluated oligoarginines and oligohistidine as the cationic component that binds to the DNA. But in the interest of time, I can't highlight each one of them, but if you're interested, here are some selected publications. So I'm going to shift gear and focus on our work in my independent career at USD. So I got really interested with cationic polymers due to my work as a postdoc, and then just because this cationic polymers has a lot of diverse application, both in biomedical and industrial fields. In addition, this cationic polymers typically bear an amine functional group, which can be used as conjugation sites or attachment sites for different small molecule drugs or even bioactive molecules such as peptides and proteins. So here are some more additional work in literature that have utilized, that have shown the utility of this amine-based cationic polymers using DMAPMA. So this is your dimethyl aminopropyl methacrylamide, I'll call that DMAPMA, and other amine-based monomers. So for example, Narain and his group from University of Alberta developed this cationic glycopolymers containing DMAPMA, a morpholine monomer, and then a sugar derivative monomer containing galactose, using aqueous RAF polymerization for siRNA delivery. Another work where some people here in the group might be interested was done by Morgan and co-workers in University of Southern Mississippi, where they prepared low molecular weight amine-based cationic polymers using aqueous raft for antimicrobial activity. So what they did was sort of like a structure activity studies where they evaluated the different cationic structures for biocidal application. So I should mention, let me go back to that slide. What I wanted to highlight on this slide is the synthesis. So in both of these studies, the preparation of these polymers, so for example, this step right here to form the polydimapma polymers takes about 4 to 6 hours to prepare. Now, in a research institution where graduate students and postdocs work 24/7, sometimes literally, these conditions are fine. But in an undergraduate laboratory setting, we need to carefully plan our experiments, and ideally we wanted to have shorter reaction time. So the goal of our work is to then develop an efficient raft methodology that will allow us to synthesize different cationic polymers. That will allow us to incorporate different monomers and produce different architecture. And to do that, instead of utilizing or using conventional heating, we're gonna employ microwave heating. So we're gonna use APMA and DMAPMA as our initial, or so we used APMA and DMAPMA as our initial test cationic monomers in the polymerization, and we use this azo-based as the initiator, and then this CPP here as our chain transfer agent. We're not the first group to employ microwave in raft polymerization. So for example, Perrier, who's actually one of the developer of raft, has employed raft-mediated polymerization of these monomers, MMA, MA, and styrene, and they have shown a rate enhancement of in polymerization for these monomers under microwave heating. So basically, higher conversion at shorter time. What they have also shown though is that these non-polar monomer did not increase in terms of its reaction for raft. Also, another group, Summerlin in University of Florida, has also employed microwave assisted raft polymerization using this monomers. But what I just wanted to highlight is that they did chain extension, or they were able to elongate the polymers using microwave raft. So as shown in this chromatogram, based on the change in retention time, they were able to extend and increase the molecular weight of the polymers with low dispersity, or dispersities close to 1. So encouraged by these results, and after looking into or searching the literature and finding out that the microwave raft polymerization of APMA and DMAPMA has never been reported, we were further motivated to pursue our work. So this is the workflow that my students follow to prepare the polymers and analyze them. So just really briefly, students would prepare the polymer stock solution by adding the monomer, the initiator, and then the chain transfer agent. They would aliquot it in different solution for time point and for time point studies, and evaluate it at different mode of heating. The reaction has to be done oxygen-free, so it has to be purged with nitrogen. Half was taken for microwave heating, the other half for conventional oil bath heating. And at certain time intervals, reactions were stopped. A small amount were taken for proton NMR analysis to identify monomer conversion, or to know how much of the monomer has been incorporated to the polymer. We then purify the remaining solution to remove any unreacted monomer. And then finally, we characterize the purified polymer again by proton NMR to determine the degree of polymerization, or how big our polymers are. So The NMR that you see on the top is the spectrum for the stock solution at T0. So as polymerization progresses, you will see broad peaks showing up, which is an indication of polymer formation. Also, as polymerization progresses, you will see a decrease in the peaks at around 5.4 and 5.8 ppm, which corresponds to the alkene protons of the unreacted monomer. Also, as polymerization progresses, you will see this broad peaks that corresponds at around 1 ppm, and it corresponds to the methyl peaks of the monomer repeat unit that is now part of the polymer. So we compare the integration of these two sets of peak to be able to identify our monomer conversion as well as the kinetics of the polymerization. So like I said, after purification, once the monomers are removed, so now we don't see those alkene protons from the unreacted monomers, we see this really nice NMR spectrum of polymers, all broad peaks, but they are still informative that will allow us to identify the degree of polymerization. So as you can see here, this peaks at the aromatic region somewhere between 7.2 to 8 ppm corresponds to the protons in the benzene ring, which is also found at the end group of each polymer. As mentioned earlier, you have these broad peaks at around 1 ppm, and it corresponds to the monomer repeat unit of the polymer backbone. So by comparing the relative intensity of the end group protons labeled as A, and then to that of the methyl protons of the repeat unit labeled as B, we can then calculate for the degree of polymerization, which is N right here. So there were a lot of trial and error done, but we finally saw promising results. So the figure that you see on the left shows the comparison of the RAF polymerization for APMA and DMAPMA under microwave and conventional heating. And as we can see that the microwave mediated polymerization for APMA and DMAPMA under microwave showed rate enhancement. So you can see that at T24 minutes, we were able to already show somewhere between 70 to 80% monomer conversions for both monomers, whereas for conventional heating, we only observed somewhere around 20% conversion. Also, we were able to plot the kinetics of the RAF polymerization, and I've shown that with microwave heating, it maintained a linear first order kinetic, which is actually a characteristic of RAF polymerization. So RAF polymerization is considered a living polymerization, which means you can stop the reaction at any point of time, and then you can take those polymers and then start the polymerization again to extend your polymer or to elongate your polymer. And this feature, I'm so sorry, why is that? And this feature is highly dependent on the fidelity of that end group right here. So to show the end group fidelity or the end group retention, we then extended our previously synthesized polymers. We did another round of microwave RAF polymerization with these polymers using the same monomers, and what we have seen is definitely chain extension. So based on this chromatogram, it is basically a gel permeation chromatography similar to size exclusion. There is a change in retention time, which indicates an increase in molecular weight for these polymers before and after chain extension. So 1 corresponds to before extension, and then 2, it's after extension. No, why is this going? So another thing I wanted to highlight on this table is that the molecular weight that we calculated from monomer conversion, the molecular weight that we calculated from the degree of polymerization, and then the molecular weight that was obtained from GPC are all in agreement to each other. So this validates the proton NMR analytical method that we're utilizing in the lab. And most importantly, all the polymers that we have synthesized in through microwave raft have dispersities close to 1, which means all the polymers that we are synthesizing have this narrow molecular weight distribution. So in summary, I hope I have convinced everyone that cationic polymers are important macromolecules with a lot of different applications, and that microwave raft polymerization is actually a powerful synthetic tool that will allow us to synthesize different types of cationic polymers. So we envision that this microwave-mediated raft methodology that we have in the lab can serve as a platform in synthesizing more complex polymers with unique and interesting architecture and properties. So for example, we can now use this more complex peptide-based monomers and prepare more complex polymers using this methodology. With that, I would like to thank all my wonderful students who have contributed in this work. Special thanks to Jonathan and Sydney, who actually moved this project forward. And then special thanks to Alex Prosnitz from University of Washington, who run our GPC samples, and then NSF for NSF for student support. And thank you everyone for listening, and I'd be happy to take any questions.
So thank you very much, Joan, for sharing your work from University of San Diego. And I know that like working with undergraduate students at the same time, publishing a manuscript out of their work is a milestone. And so therefore, thank you very much for that. So at this point, we are going to have an open forum. So first, we would like to open the floor for Bernadette. Bernadette, are you still there?
Yep, I'm here.
Okay, so for the information of everyone, it's already 12 a.m. in the East Coast, and we really appreciate Bernadette's patience in staying with us in the middle of the night. And so, Bernadette, here are the questions. So the first question came from the president of the PAASE, Dr. Giselle Concepcion. So her question is, are antibodies that target tumor-associated glycoproteins that do not get internalized considered as good potential therapeutic targeting agents? Or are internalized antibodies better? Is antibody internalization in any way associated with a faster development of resistance of cancer cells? So the floor is yours.
I think it depends on the mechanism of action of the antibody. For example, there are antibodies that do not internalize as fast that target glycoproteins, but they have their mechanism of action is to inhibit the ligands from binding to the receptor, and also the FC region of that antibody can elicit the immune response to target the cancer cell as well. So it just depends on the mechanism of action. For the antibody that I presented, it needed to internalize because the therapy is not the antibody, but the drug conjugated to it. The antibody just happens to be the targeting vector. And with regard to development of resistance, I'm not sure about. I don't know how to answer this question, but just that the antibody drug, the resistance to the antibody drug conjugate that I presented was due to the patients having been previously treated with taxanes. And so I think that the drug that was conjugated to the antibody also had a similar mechanism of action. And so that conferred resistance as well to that antibody.
Okay, thank you, Bernadette. And then the second question, please compare the pros and cons between MRI and PET imaging. The Philippines doesn't have an animal-based, well-established PET imaging R&D program. Could you please brief us on human resource and infrastructure requirements? So this question comes from Janelle Saludos.
Sure. I have to be honest that I am biased for PET imaging, so I can only give you the pros for PET imaging. But I think MRI can be complementary to PET, and I know that there are PET MR systems that can give you the technology to be able to look at the function and also the anatomical features of soft tissue. With regard to the infrastructure, the PET scanners, they are making them smaller now in that they can be available as benchtop scanners instead of the large machines. They do cost quite a bit. I think it's probably around 500,000 if I'm not
500,000 US dollars.
Yes, 500 to 750,000. But with the PET scanner, you have to be able to access radioisotopes, and for that, I'm not sure where cyclotron facilities are in, or if there are any in the Philippines. I'm not sure, but you might be able to get them from other countries if the half-life is long enough. For example, for zirconium-89, if there is a cyclotron facility in Singapore, you might be able to get that because the half-life is long enough.
Okay. And for the last question, do you think that other types of PET imaging agents can also be developed or for those existing be improved for other types of cancer? Will this significantly change the future of cancer therapy?
I think it's changing the future of cancer therapy now. There are PET imaging agents that are not antibody-based, but smaller molecules labeled with F18 that's becoming FDA approved. So I think it's really helpful to monitor response to therapy. But the issue is access to, Janelle is right, the issue would be the access to these resources.
Okay, so thank you, Bernadette, for answering those questions. And our next set of questions is for Joan. Joan, I know that you recognize these questions. This comes from Janelle. So the first question is, cationic polymers, whether natural or synthetic, are well documented to cross the membrane bilayer of the cell. For peptide-based polymers, we understand that these could be degraded by proteases. However, do we know the fate as well as the implication of intracellular accumulation of non-natural cationic polymers? Example products of your RAF polymerization in those that are based on PEG functionalized cationic polymers. Can you answer that?
Okay, so I'll be honest, it has been a while, but one of our, and I have to go back to verify this question, but one of the graduate students in Susie Pon's lab investigated the intracellular uptake of these materials. And one of the observations or the result was that it actually gets uptaken through clathrin-dependent endocytosis, and they actually get internalized, but the fate of that, but a lot of these cationic polymers also interacts with the mitochondria, so other intracellular organelles that makes these polymers, I should mention, slightly toxic. So I'm not sure if if I answered that question correctly or accurately, but I probably have to check a little bit more to answer the first question.
Okay, thank you. And the second question, in preparing polyplexes of cationic polymers and oligonucleotides, do you prepare a 1:1 mix, or is there a sweet spot in the mix? How do you determine this mixture?
Yeah, this is actually a great question and something that I did not talk about. So when we do our formulation and when we report our transfection studies, it's based on N to P ratio, which is the nitrogen of the cationic moiety to the phosphate backbone of the DNA. So whenever we're formulating, it's based on the ratio of the N to the P. So when we report it, it's based on those numbers. And in terms of calculation, again, it has been a while, but we do our transfection studies and we're trying to evaluate different N to P ratios. And then in the in vitro work, where we find the optimal N to P ratio, that's when we further evaluate these types of delivery system. So for example, the in vivo work that I've shown with the melittin dye block, we used N to P ratio of 3.
Okay, so thank you very much.
Doralyn, can I ask a very quick question for Joan?
Sure.
Yeah, so I really enjoyed your talk, and as Doralyn said, it is impressive to be publishing with undergrads. My question is on your method on the microwave-assisted heating. So you showed a nice plot where you can actually see the gain in time in terms of the synthesis. But is that now a standard practice in rough polymerization? And somewhat related to that, are there like side products, or what are the disadvantages if you go the matrix as microwave assisted versus like the standard heating?
Okay, so I wouldn't say it's a standard practice because there are still limited publications that utilizes microwave raft. One of the problem in microwave-induced polymerization is the instantaneous heating at the beginning of the reaction. So there's still, that is a great point, there you can still see some side reactions or side products in this microwave-mediated work. One of the questions that we were asked in the paper is actually the fidelity of that end group in our polymer, especially since we're doing this in aqueous condition and we have this basic amine nucleophiles that can actually aminolize the end group. Then with microwave heating, the question was, are we retaining the end group? So we're happy to show actually that with the conditions that we were able to develop, finding the right concentration, solvent, through microwave-mediated raft, we were able to retain the end group of that polymers.
And you have a very good polydispersity, right, in the end as well?
Yes.
Pretty cool.
Thank you.
Okay, Joan, there's this one last question from the president of the PAASE, Dr. Giselle Concepcion. So it's in the chat box. So could HPMA be linked to a tumor targeting antibody and also linked to an anti-cancer small molecule? Are there these therapies now in the market? How does HPMA delivery compare with liposomal delivery of drugs with or without antibody targeting?
Okay, so HPMA is basically just a monomer. So we utilize this monomer as a PEG mimic. So it's for stabilization, for stealth of our polymer. So in my recollection of literature, you can polymerize HPMA and then conjugate it to different small molecule drugs and would have the similar, I don't know how is it different with PEG, but somewhat similar effectiveness compared to PEG in terms of stabilization of this molecule. So I would say I have to dig in more in terms of the the liposomal delivery of drugs with HPMA, but these are hydrophilic monomers. So if you are targeting liposomes, you may have to, utilize liposomal delivery, you may have to incorporate a hydrophobic component to make it like an amphipathic system.
Okay, thank you. Thank you very much, Joan. So this is the end of our webinar, of our session on the hot stuff of the future luminaries in chemistry and biochemistry. And thank you very much for particularly with our speakers who have been for sharing their expertise and time about their work and the science that they do, and also for sharing those knowledge to our participants, particularly for those who stayed with us for the last 4 hours. So in summary, we were able to tackle 3 different areas in chemistry and biochemistry. That is, first, with the genomics and synthetic biology and metabolomics, we talk about more on the microbes and how they are used as a source of different biotechnological applications. And then second, which were presented by Dr. Cheryl Andam, Dr. Emzo de los Santos, and Dr. Joaquim Marques. We also were able to hear about the story about the gene that triggers asthma. This is in the topic of biosystem interaction that was presented by Dr. Ronald Panganiban. And also we were able to hear the story about using nanoparticles to potentiate the effect of antimicrobial agents that were delivered by Dr. Ruby Solian. And lastly, we jumped to the drug delivery and biomarkers, where we use chemistry and biochemistry to understand and also to diagnose cancer therapy, and also to provide vehicle to deliver different types of therapies, such as gene therapy that were delivered by Joan from the University of San Diego. So in the end, we were able to provide a very good platform for everyone. I know this is an information overload for most of our participants, but I am sure they were all very inspired about seeing this young scientists, now Filipino American, that are now doing great work in the US. And I hope I can see you here, and I would like to invite everyone to become a DOST Balik scientist. We are very happy to host you here at USA Iloilo, and so we will work on that. And I would also nominate all of you to become a PAASE member. So before anything else, I would like to thank Dr. Giselle Concepcion for spearheading these 3 weeks of sessions and different aspects of science, technology. And also I would like to thank Richmond Pancho for helping us to come up with this seminar series. And so here is the flyer to become a member of the PAASE. And so I will definitely contact you, our esteemed speakers, to be one of our PAASE members. So when we were in the States, we heard about PAASE and were able to join some of their meetings in the US and here in the Philippines. So with no further ado, let's give a round of applause. There's a reaction here. Give a clap. I would like to see all the participants to give their reactions. So to clap everyone for the job well done. And thank you very, very much. And we're looking forward to hearing more of your exciting and amazing work in chemistry and biochemistry. So good night, our dear esteemed speakers, and to all our participants. Thank you for staying with us, and have a productive day to all of you. Thank you.
Thank you.
Thank you.
Yes, Doralyn.
Yeah.
Thank you again, Ate Dorothy. Thank you for your
Oh,
commitment.
sorry.
We'll keep in touch.
Bye for now.
Yes.
Thank you guys for the